for the internet. Sometimes you have to wait for the internet to help you out. Hello, everyone. This is This Week in Science. This is the live recording of the TWIS podcast. If you're joining us live, we're so glad that you're here. Just remember, if you're watching this in video, that means this is the completely uncut, unedited version of TWIS. If you like the nicely edited, a little bit shorter version, then make sure you sign up for that podcast of ours, because that's, that'll be out after the show. Okay, are we ready, everyone?
Mm-hmm.
Yeah, there's a bunch of mm-hms and knowing nods. Okay. All right, everyone, going live in a 3, a 2. This is TWIS. This Week in Science, episode number 793, recorded on Wednesday, September 30th, 2020. Better know a virus. Hi, everyone. I'm Dr. Kiki, and tonight on This Week in Science, we will fill your head with Neanderthals, consciousness, and disemboweling. But first...
Disclaimer. The following program contains civil discourse. That's the thing where one person speaks while others listen, and then that first person stops talking and listens as somebody else talks. There may be occasions where more than one person is talking at once, largely due to enthusiasm for the subject matters being discussed, but we will try to keep that to a minimum. A informal, loosely formatted, and completely unmoderated conversation is expected, and while no ground rules have been agreed upon, at no point will you hear anyone attack anyone else's character, or belittle their intelligence, or besmirch someone else's family members. Because even without rules and moderators, people can be well behaved and interact in a civil manner. Like adults. Or children, even. They're pretty good at that, too. We choose to be civil. We choose to be civil with one another on this podcast and not say the bad things. Not because it is easy, but because it is hard to listen to people talk when they aren't being civil. So all you really need to do, it's really actually easy to be civil. All you need to do is listen to This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back, and yeah, let's be nice to each other and talk about science.
Yeah.
I mean, I do have to say, it's one of the reasons I love coming back to this show week after week, getting to talk about science and have fun with my friends doing it. It's just really, really wonderful. And I hope that you all keep coming back for the fun and civility and the science Yes. All right. What is on the show tonight? I have some stories about trust, a helpful virus, and bird brains. Because you know I love them. Yes.
I know you love that.
Yeah, wait. We also have an interview. We have a guest. Hello.
Oh.
Yes. Hello. We have a guest. We're going to talk about viruses tonight with our guest, Dr. Efra Rivera-Serrano. And now, Justin, what did you bring?
I have Adding Up the Universe. I have Vape Juice Hot Take. How nature finds a way. And Neanderthals.
Nice. Always love the Neanderthals. Blair.
I love you love brains. I love Neanderthals.
Yeah, you know what I love?
What's in the animal corner?
Just some inspiration and also some fireflies or lightning bugs, which I'm obsessed with. And then I also have a really quick story about climate change.
About what?
Climate change, you know, is this little thing. It's something to be concerned about. It's like not the number these days, but it's kind of important. So just a little something.
A little bit,
yeah. All right. Well, with all these stories and a wonderful interview coming up, I would like to remind you, as we jump on in, that if you have not yet subscribed to TWIS, you can find us all places that podcasts are found: Spotify, Spreaker, Pandora, radio.com, Apple, Google. Just look for This Week in Science. We're also on YouTube and Facebook and Twitch. You can also find information about the show at twis.org, TWIS.org. All right, so now let's dive into some of that aforementioned science. Want to talk about trust?
Mm-hmm.
Uh-huh. Well, let's talk about trust in science. How does that sound?
Yeah.
I trust science.
I trust science. And, well, good news. A new Pew report, a research survey. Pew, pew. Yes, it's a great institute that has done many surveys over the years, and they survey public ideas how public feelings about trust. The new survey is out now. They looked at 20 global publics. That means they went to a bunch of different countries. They surveyed
Wait,
more than
other planets.
Yeah, no,
Sorry.
just different countries, looking at how different people in different countries look at science or how they think about it. They surveyed more than 35,000 people to find out those public attitudes on science. So let's talk about us. I mean, the U.S. Yes.
Oh, see, that's the one I was afraid of.
Is that like Us Weekly or US Weekly? I never can tell.
It's sad, but that's the one. When you started talking about this, I'm like, oh, hopefully they left this out. If you or at least did one where if you don't include the United States, it's looking great.
Actually, and let me tell you, actually, it is overall looking great. Public trust in science in the United States is looks like it's on a slight upswing, but it's not huge. I mean, it's always kind of been around 40% or so of the public. And we sit at 38% of the US public who have a lot of trust in science. As a group, about 3/4 of the United States, 77%, have at least some trust in scientists to do what's right for the country. At least some trust. And this is great because the views here in the US are pretty similar to other global publics everywhere else. We're right around the median, actually, for the amount of trust in science or scientists.
We're not the worst.
We're not the worst. No.
In something.
Yeah.
Right. Exactly. Where it breaks down is in the area of politics. And when you break Americans down by partisan identification and political ideology, 67% of Democrats have a lot of trust in scientists. 17% of Republicans do. So the ideological divide is huge. This left-right ideological divide is really strong in the United States, but it's not alone. Australia, Canada, the UK, Germany, Italy, many other countries also have similar political divides.
Really?
The one thing that Democrats and Republicans are aligned over, however, is the value of being a world leader in science and the state of the United States in scientific achievements. We want to be the best. We think
it's the thing we don't trust.
And everybody's okay with that. and across the board, the two groups value investing money into scientific research. So there are, I think the take-home message is there is trust in science. So the war on science is not as broad as you might think. However, there have been ideological divisions that have been played, certainly, and taken advantage of. And so there's work to be done in terms of science communication and building that trust on across the board. Anyway, take home. That's my take-home message.
It was looking at the Rice University moonshot speech by Kennedy, as I occasionally refer to in my free time. And there's an interesting comment in there where he's really talking about, science leading this next step for the United States as being a country that's going into this new knowledge, and as we gain this new knowledge, we're going to be showing more of our ignorance, and by exploring these new fields. One of the interesting things that caught me there is he says that today there are more scientists alive in the United States than at any other time in our history combined, and that it is doubling. The statistic, he might have made it up, but I think I kind of trust him. Was doubling at a rate. The number of scientists working in the United States was doubling at a rate 12 times the population growth of the United States. So there was this time when people were really on this precipice, being excited about it, and people were running in droves to become scientists. They were really teaching this next generation to prepare for a future that was going to be science-based, which is how we got here to the modern age that we are in now. We got here through science. And the idea that there could be that much of the population that doesn't trust the thing that worked over the last, 70 years, it's mind-boggling.
And it is mind-boggling, but it is where we are at the moment. But interesting point, in case anybody was wondering whether or not trust in science around the globe correlates with how much, with the prevalence of COVID-19, SARS-CoV-2 infection rate. It does not. So countries that have, like India, that have very high trust in science have a pretty high infection rate. And the United States is not alone in terms of its infection rate and trust in science. So it seems to be a mixed bag.
Well, trust and understanding are different things. And then there's
Exactly.
also with India, there's the huge confounding variable of population density. So.
Population density and infrastructure and things like this.
Yeah. So the trust is there, but there are still public health issues at play. There are a lot of variables at play for sure. But good news, ish. I mean, there's work for science and science communicators to do for sure.
So I couldn't see that. I couldn't see that chart. I don't know if you still have it up, Kiki.
Can get it up.
But if we only counted, Blue America, where are we? Are we ranking higher? Are we up towards? Are we at the top? If we just ignore Red America, we're at 68, right?
Right, 67%. So
which would put us, if we only counted.
Well,
I'm just looking for a brighter spot to.
All right. Where are those infections happening, I guess?
If we only count the blue states, we're actually doing fantastic. We're number one again. Just saying.
I seem to remember somebody saying that we should only count some of the states. Anyway, while we're on the subject of counting, how much matter, Justin? How much matter is in the universe? I want to know. Do we know?
We do. We have the most accurate count. Scientists at the University of California, Riverside, they just finished a very exhaustive task of accounting for every bit of energy in the universe.
Let me guess. It's something to do with Avogadro's number.
It's California, so I'm sure avocados got involved at some point.
Sure.
But according to, this is an astrophysical journal, the team determined that matter makes up 31%, 31.5 give or take 1.3%, of the total amount of energy in the universe, with the remainder consisting of dark energy. So 31%, that's not a bad number. This is quoting lead author Muhammad Abdullah. He says, to put that amount of matter in context, if all the matter in the universe were spread out evenly across the space, this is the physicist creating the spherical cow that we've talked about.
Right.
Just take the matter, spread it out across the entire universe. It would correspond to an average mass density equal to only about 6 hydrogen atoms per cubic meter. So it's a very undense universe still. However, since we know that 80% of matter is actually dark matter, in reality, most of the matter consists not of hydrogen atoms, but rather a type of matter which cosmologists don't yet understand. Abdullah is a graduate student at UC Riverside Department of Physics and Astronomy. So of the 31.5% of the universe's energy that is from matter, only 20% of that is the stuff that we can see and interact with. So very small amount. Now we're down to that one hydrogen atom per
square meter, right?
Yeah. So one of the ways that you can determine the amount of matter in the universe is the number of galaxy clusters. You look at those and you weigh them. Higher percentage of matter would result in more clusters, Abdullah says, but it is difficult to measure the mass of any galaxy cluster accurately because most of the matter is dark and we can't just look at it with our telescopes. To overcome this, the team developed the gal weight system, a cosmological tool used to measure the mass of gals at a county fair. Step right up, ladies. Guess your weight and you win a prize.
What? Nope.
No, gal weight. Gal weight is actually short for a galaxy weight.
Okay, there we go.
A cosmological tool to measure the mass of a galaxy cluster using the orbits of its member galaxies. Quotey voice of Gillian Wilson, professor of physics and astronomy at UCR. We have succeeded in making one of the most precise measurements ever made using the galaxy cluster technique. Moreover, this is the first use of the galaxy orbit technique, which has obtained a value in agreement with those obtained by teams who use non-cluster techniques, such as cosmic microwave background and baryon acoustic oscillations, type 1 supernova, or gravitational lensing. So they created this model based on all these different looks of galaxy clusters that they could observe throughout the universe. And they put them, compared that with a simulator that said, okay, well, if we had more matter, what it would look like. If we took away matter, what should that look like? And then they took their result and compared it to a bunch of other models that had already existed. And they found, wow, it's overlapping, it's matching. So great confidence in their result, and they believe they've come up with the most accurate measurement of energy in our universe. Yeah.
I think that's exciting. I mean, it's such an abstract concept. I mean, we're sitting here in the middle, or often the edge of our galaxy in the universe, and the idea of being able to actually calculate from our vantage point the total amount of anything in the universe. It seems like a very abstract concept, and the fact that we are developing technologies to be able to image what's out there, to be able to see things better. I mean, that just it's really exciting. Getting better understanding of what's out there.
Mm-hmm.
Yeah. Mm-hmm. More avocados. I mean, avocados. Hey, Blair, you got to talk about some birds now.
Yeah, birds and climate change. It's bad news, guys.
Oh, great. Well, there's a whole universe out there, so that's
Well, Max Planck Institute of Animal Behavior and Cornell University worked together to look at temporal changes in birds. So kind of a synchronization problem is pretty common with climate change and animals that have kind of seasonal patterns. And so they were looking specifically on decades of data on weather, food availability, and breeding in tree swallows. And they wanted to see what that did to timing of when to breed and when food is available and when kind of weather takes unexpected turns. And all of those kind of things to make a happy, healthy tree swallow population, they're getting decoupled. They're spreading farther apart. So this is what happens with climate change, right? Is that it gets warmer sooner. That means that trees blossom sooner. That means that bugs who go to those trees, to those blossoms, come out sooner. And then food is available sooner. And then tree swallows will say, great, it's time to breed. But then a couple weeks later, you could get a cold snap because it's March, not May. And then all of those bugs disappear because it's cold and there's nothing to feed the babies. And the clutch can actually die. So this is kind of the big problem with this decoupling of seasonal expectations. And so they looked at the past 30 years of data, and they found that over the last 30 years, the breeding of these tree swallows would be 3 days earlier every decade for those 30 years, which doesn't sound like a lot, but it can add up. And even 10 days, even 9 days can make a difference in food availability for those babies as they're growing up. And they did find also that earlier hatchlings were at greater risk of exposure to inclement weather, which also reduced the availability of flying insects as well. So there's all these kind of dominoes that can fall as a result of laying eggs too early. And so this actually, the reason this is interesting, other than just like, oh man, birds are screwed, it also it actually explains something that we've seen historically, which is swallows, swifts, flycatchers, nightjars, other aerial insectivorous birds are declining faster than mathematical models would expect in North America and Europe. And this is probably why. It's because of this kind of exacerbated impact on the population because of decoupling of these environmental cues. So kind of having babies earlier in the spring, creating cold snaps, less availability of food, and that creating less survivability of chicks. So this can help us predict what can happen to these species in the future. And it's just another reason to kind of turn our sights back onto climate change. So, it's a good reminder when we're in this COVID panic all the time that other things can kind of fall to the side, but especially with an election coming up in the United States, it's a good reminder that climate change is ever present and the decisions we make now can affect birds in 30 years. So just a reminder.
Yeah, because I think we're not going to be able to have enough bird training programs.
No. And we also can't go grab all the eggs and put them in an incubator and then release them back to the wild. There's just not enough of us. Yeah,
the human intervention on the animal side is limited. So yeah, we got to act on the climate change side.
Yep.
Yeah, we do. Well, speaking of, not climate, just the earth, we have other issues, plastic being among them. We love our plastic. There's a lot of it on the planet. How are we going to get rid of it? What are we going to do? Well, last year, or 2018, actually, there was an enzyme, an enzyme that was discovered in a bacterium that naturally degrades plastic. This enzyme, PETase, because of PET plastics, the PETase is the enzyme that breaks down these polymers of plastic. And what these researchers, just publishing in the Proceedings of the National Academy of Sciences, have found is that when they mixed PETase with another enzyme called mhetase, which is a monomer hydroxy blah 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 something ase, and they found that they doubled.
No, that's actually the name. They got lazy in the middle of defining it. Just blah blah. It's fine. And then they did the experiment. It worked. They're like, we're stuck with the name.
Yeah,
all the documents.
No, they're not. So when they added the second enzyme, doubling the enzymes doubled the fun, or actually doubled the digestion speed of the plastics. So what they found is that they were able to speed up the whole digestion process and what they are hoping to be able to do with, here we go, the PET polyethylene terphthalate deconstruction that is added to the mono 2-hydroxyethyl terphthalate. That's the MHET, this double enzyme system. There we go. I got it. That by adding them together, that they can hopefully develop a plastic recycling system where we will be able to take types of the PET plastics, take them, turn them into PET monomers, into monomers of plastic, and recycle them into other types of plastics. So that's the idea. Someday, recyclable plastics, and hopefully, according to research like this out of the Center for Enzyme Innovation at the University of Portsmouth, we will get there.
I think that's an important distinction just that I want to mention is every time we talk about digesting plastic, I feel like we're still talking about breaking it down into tinier plastic, which in this case, it's the tiniest I think we've discussed because it's getting down to the monomers, which is really, really great.
Yeah, because those are the little building blocks of those plastics. It's not quite the same as the polymers, which are much more destructive to the environment. Possibly.
Yeah, but there's still things that we would then turn into new plastics. It's not like it's brought down into kind of natural components that we could just release into the world.
Yeah, but and it's not like taking a cheese grater to the plastic, and like a finer and finer and finer grater and just making smaller and smaller and smaller polymers. It's taking it to it's the building blocks. So it's like, okay. And if you're a 3D printer, somebody who likes to make things, you could make something, degrade it, recycle it, and then make something again if you have these monomers that are available to you. Yeah. Justin,
cool.
You got your hot take now?
Yeah, the hot take. So this is results of a vaping study have shown that significant lung injury can occur, but it might not be the vape juice's fault. So back off. Back off the CBD gummy-gasm. Don't blame the cosmic cotton candy THC. It might not be the ice cream nicotine cookie dough oil that has been causing severe lung injuries because the materials used in the heating elements of some e-cigs and e-cig devices contained nickel chromium alloy heating elements that are now being thought to be the culprit of all the severe lung damage that we sort of saw spike in 2019.
Nickel chromium sounds like a pretty bad combination if any of that is something that you are inhaling.
Apparently. So the findings, they say... I wouldn't know.
Sounds like a really bad band.
Honestly, if you describe the material makeup in this form of anything we ingest or interact with, it all sounds bad. Until you're like, oh, okay, that's salt. I get it now. It sounded like something really gross. Anyway, the findings were consistent in this, with or without the use of nicotine, vitamin E oil, THC oil, which had been previously thought to be contributing to these respiratory problems. The early results published in the Journal of American Heart Association by researchers from the University of California, Irvine School of Medicine and the Huntington Medical Research Institute were observed during a much larger study where they were exploring the effects of vaping on the cardiovascular system. While they were conducting this experiment, they observed the associated lung injury immediately after changing out their vaping devices. So it was a bit of a fluke that they even discovered this because they were doing this research and they actually weren't this is a year-long study, they weren't recreating the severe respiratory problems that people had been experiencing. At some point, the device that they were initially using to do their testing went off market, and they got a substitute device that was, and immediately they saw there was. Let's see. Within an hour of beginning an experiment, we observed the evidence of severe respiratory distress, including labored breathing, wheezing, and panting, says Michael Kleinman, PhD professor of occupational environmental medicine at UCI School of Medicine. After analyzing lung tissue from subjects in the study, we found them to be severely compromised and observed other serious changes such as lung lesions, red blood cell congestion, obliteration of alveolar spaces, and pneumonitis. I don't even know that was a thing in some cases. But
lung problems.
Lung problems. So, but what had happened is the old heating element that they used was stainless steel, no problem. The new one had the nickel chromium alloy, immediately problems in their test. One of the interesting things is, so there's still, there's still warnings on the vaping isn't really necessarily all good for you. It's not what this is
Putting
saying.
anything into your lungs is, other than just air, isn't good for you. Oxygen at its base level is rusting you, but your lungs, they help you exchange gases. Yeah. But anything, you're in smoke or particulate, anything in your lungs is not good.
Exactly. So this is definitely not you should do this, it's safe.
No.
However, if it's the thing that you're doing to combat being a tobacco smoker, maybe there's hope that this can be a thing again. Researchers aimed to study the impacts of breathing the vapors from e-cigarettes on heart function in a well-established preclinical experimental model. But over the course of nearly a year, none of the subjects exposed to vapors from the stainless steel devices, with or without additives, contracted respiratory distress, and only one of them showed a less than 10% area of inflammation in the lungs. So the preliminary studies need to be followed up with additional studies to systematically try to determine the cause of the lung problem, what's taking place with this particular heating element. But vaping by itself has been proven to cause increased blood pressure, endothelial dysfunction, and risk of myocardial infarction and stroke even. So it's not all good news for vaping. That's not what this is. However, one thing I thought was interesting is there is a condition that has been called e-cigarette or vaping product use associated lung injury, EVALI, which was recognized in the United States back in 2019 when there was people were dying from this. There were, it was kind of horrible. There was a rash of this that seemed to all hit at once, or at least hit the news all at once.
Right.
What's interesting though is that was it peaked around the same time when they changed out their device and got this new one with the nickel chromium. So there may have been an association there looking back that they're going to try to investigate as well. But one of the interesting things too, I find, is that they have said that people who vape have higher chance of contracting COVID.
Yeah.
However, the symptoms
and cigarette smokers smokers in general.
The symptoms of EVALI, the e-cigarette or vaping product use associated lung injury, mimics features of COVID-19 pneumonia. And so without testing, if you had a patient who came and presented with these symptoms, they might get classified as COVID, even if it's this, because it would look the same. It would present the same to a doctor. So that also has to be looked at. Is there a higher instance of them actually contracting, or is it this is the cause of this lung injury? And now that needs. Anyway, the study's still ongoing. This is a preliminary result, but the reason they pushed it out so quick is they wanted to give a heads up to the vaping industry, which is still a thing.
Fix your vape pens.
Hey, here's what we found. Yeah, get that out of here. Get that out of here. We're still studying, but we have such a strong hit for this heating element, it's worth taking it off the market.
And for it to be done before whoever regulates the FDA probably
you can self-regulate. You can avoid having
self-regulated industry. Yeah.
Heads up to the industry. Here's this thing we found that might improve your product and help keep it from killing people.
Very cool. Very cool. Speaking of helping and not not killing. I have a virus story. But it's not, the dreaded pandemic virus. Oh no. This is a mycofungus. That's right. Mycofungus? I mean, a mycovirus. Mycovirus. I gotta say the words right. Sorry. I will say the words.
I was going to say
live program. Words come out, not script. Blah, 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 blah. Mycovirus. That's a virus of a fungus. Okay, let's go. Sclerotinia sclerotiorum. Sclerotinia sclerotiorum. This is not
Harry Potter.
In the Harry Potter wizarding world. This is a fungus. It's normally pathogenic to plants that it infects. And or more specifically, it's necrotrophic. I mean, it's going to suck the life out of a plant. There's a study that was just published September 29th in the journal Molecular Plant, in which the researchers infected sclerotiorum. I would never get the thing to hover if I were a wizard. Oh, they infected it with a really small DNA microvirus. It made the fungus switch from being pathogenic to being a helper. The viral infection of the fungus led the fungal infection to help the plant grow faster and to improve its defenses. So it was better for the fungus. In the words of one of the authors, the fungal virus might be a good thing for the fungus because the fungus now recognizes the plant as home instead of killing it. The virus turned a foe to a friend. Which I think is such a fascinating turn of events when it comes to biological interactions. When is a fungus a friend? When is it a foe? We know that there are lots of fungal interactions in plants in which a fungus, the rhizomes in plants, they're going to help nitrogen fixation. This helps plants grow. How did these relations come to be in the first place? Did they happen because of a virus? Did they happen because of something like this mycovirus? Are there other interactions that have changed the evolutionary trajectory of fungal individuals? I think it's fascinating.
Not just fungal individuals. It's a story I didn't bring.
Everything. Yeah.
I thought it was interesting. I was about to bring, but I didn't end up bringing it. But they found that mice pheromones originated from a portion of a gene that's used in blood cells.
Yeah.
That they think a CRISPR virus might have gotten in and tweaked and did some reinsertion into the gene, and it basically led to eventually the pheromone scent that mice use to communicate. Viruses do this sort of have these interesting effects.
Did you just use CRISPR as a verb just to generally mean editing that would put a gene inside of another gene?
No, I meant literally a virus. I literally meant
like bacterial involvement.
Yeah, that broke down a virus, but it remained and reinserted.
Right.
But portions of it remained and reinserted. That's where CRISPR comes from. That's
right,
how
yes.
it. That's where we got it.
I was just wondering if that's how you were using it, or if you were using it like Xerox. Which is
no. But yeah, it's easy to explain. If you explained it long form. The bacteria have this way of breaking down by inserting its DNA. And then nobody was like, oh, that's also CRISPR? Yes, it's the same. Yeah, I'll use that word. But it's an accelerant sometimes to evolution and how it plays out in living things.
So there's evidence that mouse pheromones were developed that way. Fascinating.
Yeah, they somehow track the origin because the sequence of the pheromones is a sort of grabbed from a portion of how the gene that makes the hemoglobin, and it repeated in this weird way, and then it had something to do with also using a pathway that was once used for venom, and then also an ancient ancestor. I mean, it's like this whole hijacking of of junk DNA that the mice ended up with a pheromone, ended up stinking. And hey, we can use this. This means home. This means friend. I know that smell.
Now you're my friend. All right.
Yeah.
Oh, thank you, biology. Your complexity is beautiful. If you just tuned in, you are listening to This Week in Science. If you're interested in a TWIS shirt or mug or other item of TWIS goodness, head over to twis.org, click on our Zazzle link, and browse our store. We have all sorts of TWIS items available for you to enjoy, and that will also help support the show. So you can shop knowing that you're helping us out. Now I would love to introduce our guest tonight. Dr. Efra Rivera-Serrano is a cellular biologist. He's interested in viral host interactions. He left the laboratory as of today for adventures in the world of science communications and is also working as social media specialist for American Scientist. He also has many dogs.
I do.
Yeah, welcome to the show. Thank you so much for joining us tonight.
No, thank you so much for the invitation. So really excited to be here and talk about my favorite topic, which is, well, literally it will be dogs, but after dogs, it will be the viruses. So,
okay.
We're not talking about dogs today. But,
not today. We can talk about dog viruses. No, we will talk about the viruses. So how did you get interested in viruses? Where did that interest come from?
So I think this links back to the last story that we were talking about, because I actually started my journey studying plants. So I started grad school with the thought that I was going to get a PhD in botany. I fell in love with plants in undergrad. First, I wanted to go to med school, and then I fell in love with plants. That didn't work out, so I left with a master's in plant cell biology. And I didn't want to leave science just yet. So I started networking, seeing what my other avenues will be. Leaving a program and then restarting again in a different field was definitely challenging, but I was lucky enough to be welcomed by the virology community. And that's really what I love about most of the virologists out there, that they're just very welcoming and they're willing to teach you their way. So I joined a PhD program in virology. I was fortunate enough to just have that easy transition from studying plants in a herbarium to studying viral infections in the heart in mice. And so I loved it at first. I thought it was, actually, to answer your question, I started rotating in different labs, and that was my first one. It was a virology lab. And I just canceled all the other rotations in grad school, and I decided that was for me.
Awesome.
When I was attending meetings, the community was just so welcoming that I felt like this is my place. So then I continued studying viruses for my post-PhD experience a couple of years later and until today. So
that experience of going into rotations when you start graduate school, some graduate programs require you to do rotations, and others you can come in and just work directly with an investigator. I always think that those rotations are really important, because you don't just learn a different skill, you're getting a different window into a different area of science.
Yeah, definitely. And to me, again, like I came to study viruses with a mentality of studying cells, albeit in plants, but just how cells work. And that was my background. And just, again, as we're talking about virus-cell interactions, just switching your mentality and seeing infections from the cellular side and less so from a virus-centric side, and being able to switch those around gives you a different perspective, and just like any issue in diversity in real life.
Yeah. So let's talk about that for a second. The perspective of from the virus. We know, okay, there's a virus. It's going to attack the cell. It's going to get into the cell. It's going to use the cell to replicate itself, and then it's going to kill the cell most often. how do you flip that around and look at it from the perspective of the cell itself?
Yeah, so you can imagine. So when I'm teaching this, I always say, like, imagine your cell as this is your house, this is your, your happy place. And if someone is trying to break into your house, I always hate the story from a virus-centric side, that the virus just comes in and does all this and the house explodes. But in reality, there's a lot of things that are in your house that are protecting you, right? We have walls. Some people have alarm systems. We have weapons. And the cell is no exception. So I think the model of a virus comes, does this, explodes, and moves on is a very simplistic way. When as soon as a virus enters a cell, there's a whole lot of things that the cell does to try to prevent that. The cell is not a bystander; it just reacts. So I spent a lot of years just studying those quote unquote canonical pathways. So like things that essentially almost cell in our body does, that is not special to our immune system, but how our cells really responding. And there are differences in different tissues and whatnot. But at the end of the day, if someone is trying to break into your house, there's a lot of things that are going to happen. You're going to have an alarm system. You're going to call 911. You're going to call for help. Maybe the neighbor's going to come help you, and the body's no exception.
Except that sometimes the virus gets in, and then it's just there. You walk past the living room. It's there on the couch, and you're like, okay, you should go. I think we're gonna stay.
No, but
by the way,
again.
By the way. And I'm gonna clone myself like 100 times this morning, and so I hope you have enough coffee.
But again, I think in biology that's also another issue that we face. That is that we tend to think very individualistic. So that same burglar, you might not be able to stop it the first time. Maybe it came and robbed your house, but if you can let the neighbors know and call 911, you're preventing it from infecting others. So sometimes it does happen that the virus comes and it infects it. And it actually is almost always, because that first infected cell, if it's not able to, it's very unlikely that it will just stop the virus right in. Almost always what it's doing is preventing it from spreading. So that burglar might be breaking into one house in the neighborhood, but the whole problem, the key in biology is to protect the host. It's fine if you lose one cell. It's not so much if you lose a whole tissue or an organ.
Right. So what kind of differences have we discovered in different tissues in how they respond to viruses? Is it like the same across the board? They just, this is I'm the liver and I respond this way. I'm the heart and I respond this way. Or is it virus specific as well?
So you can have any different permutation that you can come in a matrix. There's a different something different that can happen from a virus perspective, from a cell perspective, and not just in an organ. Like a liver has different cell types, and each one of them behaves differently. So and that relates not only to their genetic makeup, so like what proteins do they have, but also their kinetics. Like some organs respond really fast. So you can imagine there's a lot of different. Our body, just like as we're seeing here in the image, we can get exposed by microbes by essentially every route that you can imagine. And right now, as I'm sitting here, I'm being exposed. There's dust coming on my skin. My skin itself is a barrier. I'm breathing in. There's stuff coming into my lungs. And so this is what happens essentially all the time. Every time that you breathe, every time that you eat, this is happening. But almost always our body does a really good job, and that's the part that we sort of neglect. We tend to focus on the time when we get sick, but not every day that we're not, because our body is really good at it. So it's not the glass half empty, half full, depends on how you see it. But yeah, to answer your question, there's viruses are very different from different families. Their genetic material is different. Our cells are different based on how the virus is in its genetic material, whether it's DNA, whether it's RNA, whether it's circular, whether it's linear, all those determine the type of response that our cells will have. And so our cells essentially have evolved in ways to recognize anything that our body would not normally produce. So if you come with a virus, for example, and I have here a cartoon, you can imagine a virus sort of like, I tend to imagine a genie in a lamp where that genie is protected into a protein shell, and the genie being the recipe to make more viruses. And so it cannot really, it needs a trigger for that genie to come out of the lamp, and the lamp being what we call the capsid. And so viruses have evolved to release that genie only once it is inside the right cell. But once that happens, our cells just know, okay, the genie should not be here in the cell. Let's start trying to stop it because we know what's coming. And so that's sort of like the general thing that happens. And as the genie just tries to make more copies of itself, our cells at the same time are trying to stop that, not only again from that initial infected cell, but also alerting neighbors within the tissue, like there's something going on within me, you guys need to be prepared. So then all the other cells, neighboring cells, start priming themselves and getting ready. So again, going back to the neighborhood analogy, the alarm system is going off, it's loud. So my neighbor knows that he needs to start locking the doors. There's something around. So that's sort of how the thing works.
Justin, are you muted?
Sorry. What is this mechanism of intercellular communication, and they're not and is the defense against the genie, or is it against the lamp itself?
So most of them is really what they're sensing is patterns in the genie. So for example, for a coronavirus that has an RNA genome, there's what we call like patterns in its nucleic acid that our cells have different proteins that say, okay, this should not be in our RNAs. Therefore, this is a foreign material. Let's start activating a pathway. A DNA virus has different features that our DNA does not have. For example, if you have a virus that has a DNA genome, when it enters and that DNA is in the cytoplasm of the cell, our DNA should not be in the cytoplasm. So that by itself is a trigger, and our cells like, why is this happening? And so that's really how it works. And to answer your other question, most of these are based on cytokines and chemokines, which are very small proteins that get secreted out of the cell. So you can imagine this as like signal flares when you're in a boat and, like Titanic kind of thing, and you're just like doing a signal flare. That's really what the cell is doing. It's just launching and releasing little proteins that will then bind to neighbor cells. And they will respond to those signals as, okay, if I'm sensing this was released by an infected cell, I need to be ready. There's something around this area that should not be here.
That's interesting because, so with the flu, we know that there is a cytokine storm that can occur that is kind of the bad side, the downside of the cytokines, and in what they're doing is leading to an increase in inflammation, and that inflammation is an increase in heat that could potentially denature that the viral DNA that could, is part of that whole defense system for the tissue, for the host, right?
Yep. Yeah, correct. And again, it's like no different. Like you can start blasting an alarm right now. There's someone coming into my house, and for the first 10 seconds, you'll be fine. Okay, I got the message. But when that siren goes on and on and on and on, like clearly no one's really going to enjoy that because there's a lot of after effects. So it's a lot of timing that has to be regulated, and that siren has to be turned off. You cannot just have that alarm going on all the time because you might not be able to hear the neighbor. You might not. So there's a lot of complications when that misregulation goes or regulation goes wrong.
Yeah. So we've got the genetic material that wants to get into the cell so it can replicate itself. The capsid is that protein sheath around that genetic material. And sometimes viruses just have the capsid and the genetic material inside. But this doesn't look like the SARS-CoV-2 virus that we've seen pictures of with all of the little antigens sticking out the receptors
Crown.
sticking out, yeah, the outside of the cell.
Yeah.
What's going on there?
Yeah, so you can have the two different varieties and sort of like an intermediate too. When you just have that capsid, which is just a protein shell composed of proteins from the virus, and inside the inner material, that is what we call a naked virus, and that's why I'm on Twitter like naked capsid. That's essentially as simple as it gets. But a lot of viruses also have an extra layer. Some even have multiple of lipid fat, what we call an envelope, and that is derived from the host plasma membrane. So as some of these viruses, such as the coronavirus, egresses or leaves the infected cell, it gets sort of like engulfed in that plasma membrane of the cell, and so it attracts that. But in addition, as the virus is producing its own proteins, if you think about the coronavirus, it produces the spike protein as one of them, and that spike just sort of like gets into the plasma membrane, and sort of like you can imagine it as it buds. It's not taking only the lipids from the cell, but it's also taking its own proteins that were embedded into it. And so this one will have an extra layer. And then for these viruses, that's where they're sort of like, if you imagine what the virus is going to need to infect an next cell, again, if we go back to the burger analogy, you're gonna need a key and a door. So their key to enter the next house is going to be embedded into that envelope. So when the pandemic started, that's where all the discussions were. If you can dissolve that fat layer, that fat envelope, and take away the key with it, then that virus is inert, and it cannot enter a new one. So I get the question a lot of like why some viruses have it and why others don't. But I think again, it's sort of like why fish swim and birds fly. It just suits you whatever you need better. So like a virus that, for example, transmits through the enteric route, that has to go through your stomach, and there's a lot of acid there, that lipid envelope will just get dissolved, but they will be more stable for like fecal oral transmission. So they don't need the envelope, they just need stability. And contrary with a virus that spreads, for example, through coughing and through droplets to the air, they need humidity. They don't go to the stomach pH. An envelope is more suitable for their needs. So you can have essentially and like viruses like hepatitis A sort of like has an intermediate phase. So you can have viruses that have the two phases that are semi-envelope in the body, but then naked envelopes in the environment for stability. So you can, if you can imagine, there's a virus out there that does it.
So they're just basically the wonderful complexity of biology and chemistry.
Yep.
It's giving the viruses what they need. Right?
Yeah.
One of the things that has that's really interesting is modern microscopy and our ability to actually be able to see these viruses and see their shapes and what tissues they're in. And you've done a lot of this fluorescence microscopy and taken really gorgeous pictures. How do you do that? How do you find and show a virus in a tissue?
Yeah, so in order for you to really get at, here's a capsid of a virus, and here it is, you have to go back sort of like old school and do electron microscopy, which you're looking at, if you think about like coronaviruses that are 30 nanometers, you really need to have a high potent microscope, and those are the ones that you tend to see like in black and gray shade. But that's when you really want to look at the structure of the virus, and here is like I mentioned, like the crown. But there are other ways that you can visualize sort of like in a macro way, where not only like yes or no, there's virus present, but there's a lot of things that the viruses are doing to the cell. And so our cells, if you can imagine our cells as a fried egg, and the cytoplasm being the white and the nucleus the yolk, the virus is doing a lot of things to rearrange that space to suit its needs. So it's not just coming in and here's my genetic material, let's leave. it's actually making it its home. It's distributing cellular organelles. And so that's what I studied. And for that, we use like fluorescence microscopy. So cells are clear. You cannot really see them, see their inside, but you can tag them. So if you go right now, especially here that it's almost like midnight now, if I go to my backyard, it's pitch black, right? So I won't be able to see anything, but perhaps I can see some fireflies because they are producing light, and those are really easy to see. And so this is sort of like the same concept. So in here we go back to like physics and chemistry, and we're tagging different molecules, whether it comes from the virus or we artificially tag them, but we are irradiating a particular light of a known wavelength. And by just physics alone, we know that this fluorophore would absorb the light and will emit at a different wavelength. So then we can visualize that change. And so and there's different spectrums of molecules that have these properties. So some of them, like when you ignite blue light and then they release green light, or vice versa. So you can start making all these different combinations and start illuminating the cell. And so this is done a lot just to look at the cell itself, but that doesn't mean that you cannot do it to visualize the virus that is inside the cell as well. And so here's, we're looking at an example of how the microscope works. It's just a lot of physics and behind it and getting the right light, but the concept is that if you have a compound, for example, that binds to DNA and it happens to emit blue light when you shine it with ultraviolet light, because our DNA is in the nucleus, when you put that light on, the nucleus will light up. So if I put light in the pine tree that I have in my backyard and I put blue light on it, every time I turn the light, I know exactly where the pine tree is at. If I put red lights on a different tree, then I know, so I can start labeling my whole backyard accordingly. So and again, if you do the same thing and put a light on the burger that is trying to bring to your house and you give it a yellow light, you may be able to track it as it goes around the neighborhood. And doing this combination is where you can see not only where the virus is at a given time, but where is it at and what is it doing to the cell. So we have an image here of a virus that in this case is in color green. So there's a lot of different cells in the picture in the image that we're looking at, and we're tagging different things in the cell, different colors. So the nucleus of the cell is going to be blue. So that means that every little blue oval shaped thing that you see is going to be the nucleus of each cell. And then we're different components in the cell, different colors, but we're also wanting to see where the virus is at. And we tag it with green in this case. And so
that one cell looks bad.
Yeah, so the one cell in the middle is lighting green. So again, if I was to put a vibrant color into a burger and that shines light to it, I know exactly in what house he's at. So I can tell you that cell in the middle is infected. Now at this resolution, that's not a virus, that's just a whole cluster of virus that we're seeing. But it gives you a lot of information. This immediately tells you that the virus is in the cytoplasm, it's not in the nucleus.
Can you use knowing where the virus is and being able to find and identify the virus to treat somebody who's infected with virus? Yeah,
so that's sort of like the concept of taking like basic knowledge to more translational research. Because for example, if we know that the virus replicates in this particular compartment of the cell, it needs it to, that's where the replication of the genome, the gene has to be there, the gene has to make more copies of itself in this spot. Perhaps we can disrupt that spot temporarily, and then that hampers the replication process. Perhaps the virus needs to move from the cytoplasm to the nucleus, and it needs some proteins to move it. If you disrupt that, it cannot get to the place where it needs to get. So you can target that with drugs, and then the virus essentially cannot replicate. So there's many different things that that you can see, and conclusions you can make by understanding the requirements of viruses from a cellular perspective.
But then isn't there this whole other layer? That protein that's doing that transport probably has another function that was very important that it was doing. So if you're shutting down that transport protein, what else have you now affected? So this is tough.
Yeah, no, and that is part of why viruses are, so tough to treat, if you will, because their whole life cycle relies on host proteins. So there's two ways that you can target viral infections. So you can target components of the virus, so a particular protein of the virus. If you target the protein that is required to make more copies of the gene, our cells don't have that protein. It's a specific targeted approach. So the chance of toxicity is less because it's a very direct approach. But you can also target, again, the cellular side. And those, again, as you mentioned, it runs with the problem that there's a cell in our body because our cells need them. And so there's that two ways, and they're going to have pros and cons. So you can imagine a more targeted approach has the pro of it's going to be very highly specific to the virus. But that means that it's only for that virus. Whereas if you were to use a more broad approach and just say this protein that poliovirus uses, hepatitis uses, blah, 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 so you can repurpose that same drug as long as it doesn't have higher toxicity to our bodies, and you can repurpose it for multiple viruses. So it's a faster way of discovery, especially when you have emerging diseases. If you know that they belong to the same family, it is very likely that they rely on the same components, so you can just target them that way faster.
Yeah. And and that's something that's been that's being looked at for SARS-CoV-2 with the ACE2 receptor that we know is targeted by the spike protein. There are ACE inhibitors that we already use for multiple diseases that are being looked at as possible treatments. We haven't had enough of the large randomized double-blind studies done yet, but but at least we have drugs that are out there.
Yeah.
There's a question from my droid, which is not my personal droid. That's their name in the chat room. Who says, speaking of mRNA, how does mRNA COVID vaccines work, or how would an mRNA vaccine then work?
Yeah. So especially if we go back to the genetic material of the viruses, some viruses have DNA viruses, some viruses have RNA or DNA material, some of them are going to have RNA material, and those within have many different ways of exist. You can have an RNA that is in the right sense, what we call that, it means that it's ready to go and it makes proteins, or it can be on the opposite direction and then you have to retranscribe it. But for the most part, in our cells, mRNAs are made in this particular orientation that our ribosomes in our cells read and make a protein out of it. And so coronavirus is no exception. For example, their RNA is essentially a very long 30,000 bases RNA that just make a big protein that then gets cleaved into smaller proteins, and there's different ways for doing that. And so mRNA vaccine is essentially just that. You just have the recipe that when you give it to our cells, they're going to make it into a protein. Depending on the sequence, that protein is going to be made. And so if you have, for example, the sequence, the mRNA sequence for the spike protein that the virus makes, in absence of a virus, you can just give that sequence. Our cells will read it, produce the spike protein. There's no virus out there. The gene is not there. The other components of the capsid of the LAMP are not there. But it is a foreign protein what will be produced. So with the hopes that our body then will create antibodies against it because it's a foreign protein. Doesn't know that it is for this virus. It could be the spike protein from a different virus, but that is the concept. So you're giving our bodies the recipe. This is how the spike protein of coronavirus looks like, make something that if we were to get infected, you can stop it.
And by stopping it, is it going to interfere with the translation? Is it, or the printing of the virus's protein or RNA strip? Is it going to interrupt that surface so it can't get replicated, or?
Yeah, so because it's targeting, in this case, a spike protein, and the spike protein is on that lipid layer of the virus, what our body is going to make are antibodies, which are proteins that will recognize, in this case, the spike protein. So if you have a virus that has spike proteins making that crown around it, if you're vaccinated, in this case, for the spike protein, our bodies will create antibodies that will then sort of like bind to that spike. You can imagine as a neutralization. And so that if you take away all the keys from the virus that needs to get into the cell, and you code them, for example, and apply it through antibodies, then that virus cannot get into a cell, so it cannot do its thing.
Yeah, it's like
really
you're.
early.
The way someone briefly described it to me was that it is making your body pretty much produce the effect of the vaccine. So it's like when you get a vaccine normally, it's putting in that antigen that your body then has to be like, what is this thing, and recognize it and make that immune response. But then what you're basically doing is you're going, nope, this is the thing that there's the antibody right there. There you go. So your body has it already, and if it sees it again, it knows what to do.
And can we really train that much cell tissue at once? I mean, does that communicate throughout the cells, or does it have to get delivered to the cell, each cell through a drug? How does that?
So it's sort of like training our immune system. And so our immune system is white blood cells, so they're just roaming around our bodies all the time, and they create memory. And so by doing this, if you have a strong immune response, and our body learns to recognize this is spike protein, when I see it, I know how to destroy it, and it memorizes it, and that's memory immunity. The cells are going to be, they're long-lived, they're going to be circulating our bodies for a long time. And it depends, and that's why there's vaccines that are, forever, and you don't need a booster. There's some vaccines that you need boosters. So it really depends on that antigenicity and the immune response that our bodies mount to it. And so a lot of people don't understand, like, why I have to get a polio vaccine every 10 years, but for flu, this one, or, for hepatitis A, it's like 2 every 6 months. And in reality, it just depends on the antigenicity of how our bodies react to it. But in the perfect world, our bodies, our immune cells, will just memorize the sequence of a spike and parts of the spike proteins forever. So, and they'll just be roaming around our bodies. And the whole concept is that when we get infected, those cells that know how to attack it will multiply and divide, because now they're needed, and start producing antibodies to neutralize it. And once the infection goes down, then those different subsets sort of like disappear, but you're always going to have that memory in our body.
Yeah, that's what's amazing about our immune system, is the fact that it's not just reactive right here and now, that it does have that memory. And thank goodness for that.
So this is exactly sort of like, if we go back to the drug repurposing, right? The pros and cons. The system that I told you of first, of how the alarm system, right, in the neighborhood works, and how all of our cells essentially have it. Again, it's not very specific. Your alarm system doesn't tell you this burger is coming versus this one is coming. It's just an alarm system. And so going wrong, like a cytokine storm, then that's really why. It's just it's made to recognize a viral infection. The system is not saying, oh, here's a coronavirus, and here's a hepatitis virus, and here's a herpes. It's just patterns, just like a drug attacking or being a broader drug versus an adaptive memory immune system. It's a very, very specific response. So.
Recently, there's been a lot of talk about how SARS-CoV-2 affects different tissues. And we've learned that the ACE2 receptors all over the body in lots of different tissues. And one of them is the heart. And you were you have this amazing Twitter thread that you wrote about viruses and the heart. And this was, as you mentioned early on, a very a focus of your study for a long time. And you were also recently quoted in an article by Ed Yong in The Atlantic talking about what's going on with SARS-CoV-2 in the heart. Is it unique compared to other viruses?
So, spoiler alert, it's not, or at least I don't think it is, because we've known that viruses, a lot of viruses, at least over 20 human viruses, are somehow, I will prefer to use the word associated with cardiac damage, because many cases are indirect, but it's not the a standalone case. So viral myocarditis comes from itis is inflammation, myocard is from the myocardium, so it's inflammation of that myocardium in the heart tissue. Can be caused by a lot of things. It can just be genetic, or it could be caused by a bacteria or a parasite, but a lot of cases, especially in the US and Western Europe, are caused by viruses. And sort of like for the longest time, it is known, but a lot of the literature comes like from a very few labs, because not a lot of people study them. And in reality, it's like when you think about all these viruses, this is not their main site of replication. They're just like a secondary organ, or their effect is indirect. So viruses that we tend to associate with myocarditis, this effect on the heart is an indirect or a secondary effect. It's not really where they're replicating, and they're only, like, they didn't just come to our body to replicate in the heart. Right. And I always say that our heart is essential. Like, a virus really doesn't, I don't want to like humanize a virus, but it, you know, if you want to create more copies of yourself, and for a virus, the most important thing that it wants to do is to spread from host to host. If you're a virus and you just want to infect this person, you're dead. You're never going to survive in the population. And so destroying an organ that is essential is, even if you were to do it, it's probably never going to spread, so you're never going to find the virus in the population, right? And so a lot of these viruses just have this off target. Now we're seeing a lot of cases with COVID-19 patients that are coming as myocarditis positive. And so I think my whole take on this is, because there's so many cases, we are now seeing a lot of myocarditis cases. And the reason is because pre-pandemic, when you have a viral infection that somehow affects your heart, for the most part, they resolve on their own. And so you get a viral infection, some virus can get to the heart and destroy our cardiac cells, many of which are not replenishable. So muscle cells in our heart don't undergo mitosis. So like going back to high school biology, cells divide, make more copies of themselves. So cardiac muscles cannot do that. Once they are differentiated, for the most part, over 90-something%, they just do their thing, beating. So the bulk of the cells that we have in our heart right now are the exact same ones that we had when we were a kid. They're just the exact same one. And so,
I had no idea.
Yeah, so they are just doing their thing. They're differentiated, they're beating, and which comes with a consequence, right? If you lose them, then they do not come back. And so, for the most part, historically, all these viruses that cause cardiac damage, the symptoms sort of like resolve on its own. So virus comes, either destroys some cells, or our own immune system goes wild, like a cytokine storm, as we mentioned. It starts killing the infected cell, because if you destroy the infected cell, that virus cannot spread, it ends it, right? So our immune system is trying to do the right thing. It's just doing either too much or unfortunately we need those cells because they're not going to come back. But for the most part, the infection resolves, and you're never going to see it. You're never going to have a symptom, and you just move on. So a lot of the statistics come from post-mortem biopsies. So someone passed away, and they're doing biopsies on the heart, and you can find all the cardiac lesions that reflect that at some point we were infected. Again, because these cells are not dividing, because we're not replenishing, all you're going to find is a foci. This is a spot where muscle cells would have been. Now they're not. It's likely that a virus killed them, but our bodies clear the infection. It can progress to other conditions. You can even to cardiac arrest. And so, but for the most part, it doesn't. But now we're seeing a lot of cases because we're in a pandemic. So a lot of people are infected. There's an intricate relationship between the lung, where coronaviruses are affecting, and the heart, the oxygen and CO2 exchange. So you have that relationship there. Plus, now we can talk about that later. Is the age distribution that we're seeing. Historically, because it resolves on its own, and unless it's a biopsy, you don't know that you had a viral myocarditis. You never notice it. You're only going to notice if you have a symptom, and so that's usually on younger folks, and likely because they have a higher cardiac demand. It's very common on athletes because their heart is, at its max peak compared to someone that isn't. So even though you might not have a symptom right now, if you start building all those cardiac lesions over time, and then you push your heart really hard, that is why myocarditis comes at a very high or frequent cause of sudden death, because you don't know that it's there until it's too late.
That's what we need, is some way to see what kind of cellular damage we have in the heart while we're still alive. You can go in and probably do electrical tests to see how well the muscles transmit the electricity through the heart along their channels. But
and we may be getting some information on this in the coming years, because a number of NFL players, as in the past a bunch of basketball players, have come down with COVID. How does that affect? I mean, that's not just an athlete; that's an elite form of an athletic body that has been training a lifetime, working that hard to do these things. And as so often happens, sports medicine teaches us a lot about how to fix ACLs and ankles and sprains and concussions and everything else. Now we're going to put COVID on that list, I guess.
Yeah. It is, I guess it is fascinating, but at the same time, I think the biggest problem, or the thing that we need to really consider, again, this has been going on for a long time. It's just we usually just don't notice it unless it's a really bad case that you have cardiac issues or arrhythmias or whatever. But I personally don't remember any in my 30-something years of life, but I'm pretty sure that there's some cardiac lesions just by statistics alone and the number of infections that I've been encountering in my life. I'm sure there's some lesions there, but that's important to consider when you were talking about asymptomatic cases. And this population that says, well, I'm healthy enough and I'll be okay if I get COVID and I'll just have a little flu-like symptom and I'll move on. But what we need to remember is that there's a lot of secondary effects. And perhaps you were asymptomatic and you were infected at some point and you never experienced any fever, any coughing, but that doesn't mean that there was some damage to your heart, either because the virus got there or because there was some mild inflammation triggered by our immune system in response to the virus when it tried to stop it that damaged some cells. And so some studies are coming to get at that, but my criticism is that it's very hard for you to have a control group during a pandemic. So And that's important to sort of get at, because we don't know how many cardiac lesions were there prior to this one. So like, yeah, you're seeing a cardiac damage now, and you're relating it to COVID cases. But what if only 10% of your heart was damaged was caused by COVID, but you had like 30 other percent because you were exposed to like Zika virus or Coxsackie virus or whatever at some point, and it just adds up. So without a control group, it's really hard to know. But again, because a lot of viruses do it, it's sort of like you're just hammering and hammering and hammering.
That's a really, I mean, just the concept that over time there could be an additive effect of multiple viral infections, that just over time, maybe it just builds up until finally there's enough dead cells, or there's enough scar tissue that's formed that, it's not going to work right anymore.
Yeah, and most of I forgot the percent, I think it was like, it's 20 something, I forgot. It's a high percentage of, even from those biopsies, when they're trying to like get genetic material to sequence, okay, what is in that heart? It's often at least 2 or more viruses that were associated with it. And that doesn't mean that you were co-infected with both. It just means that perhaps when you were 20, you got this viral infection, and then when you were 45, you got another one, and then when you were 60, you got another one, and that's, but which one did the most, we don't know. Right. But again, these cells don't replenish, so you start, taking some and taking some, losing some, and it adds up. So, especially in this asymptomatic cases and younger populations, the chances of them, and this is something that we don't know. I hate scaring people, but we don't know. If you're like 20 something and you already lost like 10%, you still have a long way to go, and we're still gonna continue to be exposed to viruses, bacteria, parasites, right? And so,
so that low immune response for those who aren't showing symptoms might actually be incurring the same kind of damage that takes place in somebody who's getting the cough and the sniffle on the heart. We just wouldn't know.
Yeah. So again,
that is frightening.
Speaking to if we're going to just focus on the heart for just 1 second. There are viruses that can get there, like Coxsackie virus, for example. I love dogs, so like parvovirus, like that's one of the vaccines that we use for
the dogs.
For dogs, so, like there's viruses that are known to directly affect the heart, and what I mean directly is like they're doing something. They're going out there and they're doing something, destroying a cell or something. But a lot of cases, if not most of them, are because of how our body responds to it. So either because of these viruses are circulating in our blood, and long and behold, blood gets to your heart, right? It's a pump. So all those cytokines are going to get there. All those immune systems are going to go there as they're traffic to our body. So if they see something there, they'll just attack, or our cells are going to respond. So like if the cells are secreting cytokine, our cells in the heart, and they're going to the blood, our cells in the heart also have those same receptors that our cells in the lung will have, so they will say, okay, we got to react, and so it's a problem. And most importantly, we have a cell type in our body that is called the T cell. So like B cells make antibodies, and T cells are another type of cell, white blood cell, that a subset of them are made to kill infected cells, virus-infected cells. And so even if you have like a little bit of virus that gets into a cardiac cell, a T cell will kill it. Again, trying to prevent the virus from spreading, it will kill an infected cell. And you might not have a massive inflammation because that cell was very directed, but again, it adds up. So if you have another T cell killing that infected cell to prevent from spreading, and then another one, you don't need a massive inflammation. You don't need a lot of symptoms for that event to happen. And so that is a progression of how it happens. So you can have viral entry and replication. The virus is replicating in the cardiac cells, and in response to that, if the cell itself cannot just stop it through this system that we're talking about, the alarm system, that's usually the first thing that will happen. The cardiac cell will be like, maybe I can handle it on my own, But if it cannot, then an army of immune cells come and start creating an inflammation and start trying to ease the viral titer, the viral burden. And most of the cases, that resolves, and that's enough. You're gonna have some mild inflammation. You're gonna have some necrotic tissues because the cells were lost. But then sort of like you just move on. And worst case scenario is when it really goes bad and you just have massive inflammation. There's just a lot of cardiac cell loss that cannot be replenished.
Yeah, and because it's your heart, it's not, there aren't necessarily the inflammation that comes if it's mild. There's, you know, maybe your blood isn't getting where it needs to go quite as effectively, but it's probably still pretty effective, and so you probably don't really notice. You're just maybe not at the top of your game. You're just not, you know, peak performance, but you're still okay, right?
And if you have an infection, for example, that affects your lungs and your breathing is not right, but you're just lethargic, you're in bed anyway. You're not running a marathon and pushing your heart, so you might not even notice it. You can have a virus that affects your joints, like a chikungunya infection. You just don't want to walk. So if you're not walking, your heart is close to rest. You're never gonna see like, oh my, I have like arrhythmias or something. And so that's usually the part until, again, in the case of athletes, until you really notice it because you're pushing it too hard. And in a pandemic where asymptomatic transmission is happening and all that, they might not even know that happened. And perhaps the heart is still recovering because this pandemic is not over. And so even if they got an infection like 2 months ago, what happens is that when you lose those cells, muscle cells, even though they don't come back, there are other cells in our heart that take over that spot to like patch it. So like if you get a cut in your finger, right, it's going to be open and then you're going to have a scar. Some other cells are going to start like patching it. But you're going to see that scar forever, right? Because it's in a different orientation. It cannot stretch as well. It's the same thing happens in your heart. You're going to lose your muscle cells. Some other cells are going to take over, but it's not going to be the same. And we all probably have scars from when we were kids that we will always have them. And it will never be the same tissue. So.
Gosh, I feel like all our listeners after this are going to be thinking, all right, do I have heart damage, or do I have COVID anxiety, or do I have political anxiety, or is it the smoke that I'm breathing in, or is it, they're just what's wrong with me?
But I always like to say there's a reason why we don't know this until postmortem. It's because our body is really good at, you know, stopping it and moving on, and this is biology. We adapt and we move on, and so is that finding that balance of, yes, this is happening. We all need to be, and I care about this topic because it really affects the way that the young crowd thinks about asymptomatic infections and, oh, I'm good, I'm done. But I want them to protect themselves for others, but also for themselves from that perspective. But I also like to acknowledge that, again, our body is really good at coping with it, right? You didn't have any symptoms, and most of us haven't had a heart issue, even though we have had probably an inflammation in the heart at some point right now. And so it's not like we're all just going to die of a heart attack, because lo and behold, our bodies were made, we've been coping with viruses for many, many years. Our bodies, if it was that bad that one virus will kill our heart and kill us, we would already be dead as a species by now. And so,
and you know, if the way to have a healthy long life is to avoid exercising too hard, it doesn't sound.
Actually, I think I got a couple of questions like that, and I'm like, well, I mean, and especially if you don't know that you have, like, you're just going to like stop living, like that's not, especially when even after, this is before the pandemic, during, and even after this ends, this is not the only virus out there. And I just told you that a lot of viruses do it. So even after this one ends, there's still the viruses roaming around every day, you know, and so we cannot just stop living because, you know.
Yeah. Keep moving. Move as much as you can. It's good for your. We're a little meat machine, right? There's a question Blair wants to ask.
Oh, so whenever we have a virologist on the show, I have to ask, and especially since you were talking about how kind of viruses, I know you were kind of anthropomorphizing, but they don't want to kill their hosts. It's very reminiscent of parasites, of course. And so I have to ask, I don't want the textbook answer. I want your real answer. Do you think viruses are alive?
I know where this is
consider them
going.
to be alive? Yes.
I knew where this was coming. I can. Well, you really want my answer? This is a debate.
We're gonna laugh anymore. You can say whatever you want.
No, I saw it. You know, the whole debate was like on Twitter yesterday too, and I was like, well, I'm gonna stay away from this one.
Sorry.
And again, it depends on, again, you're gonna go into the tangent. Like, it depends on how you define life and whatever.
Of course.
But again, I personally think that, and this will link back to the mRNA vaccine question. Because of, if you were to take an RNA virus that has, its genome is basically an mRNA that when you put it into a cell, it has a recipe to make more copies of itself. If you just take that in principle, and this is something that we do in the lab, we can synthetically recreate a sequence of a coronavirus in the lab. We can take adenine, we can take guanine, like every single nucleotide that makes RNA, put them in the sequence that codes the virus in vitro. Like, there's nothing here but building blocks, just like one of the stories that we talked about. If you just put all these building blocks in a test tube, in the right sequence, put it into a cell, that cell will make virus. Meaning, are we really creating life? And if you were compare it to a parasite, we cannot do that with a parasite. We cannot do that with an obligate bacteria. We cannot just create a bacteria in a test tube or provide a sequence that will make it.
Yeah, tell that to George Church. He's working on it.
Not yet. But you can with an RNA virus. Like, I can just go, just like how we buy DNA sequences for PCR, and I can just buy a sequence of a virus, and that alone is going to make a virus. So, like, that's a fine line, but I'm like, if it's really life, then we are creating life in a test tube, like, from sequence alone, and I don't like that concept. So that's why I feel like I don't think they are alive, because I can buy them, like, and a chemist can say, like, this is the sequence, this is the sequence, and the cell makes it. And so that is my
But that one's temporary.
That's a nuanced answer. That's very different from the pedantic answer I usually get. That's just like, they can't replicate on their own, so they're not.
But that is a little bit.
It's more chemistry.
Time sensitive.
Yeah,
because
they're molecules.
If we get to the point then when we can design a bacterium, send it off to the chemists, and get that sequence back, and then produce it, does that then change your opinion? Or does that mean that life doesn't?
It probably would.
Okay.
It probably would, yeah.
Okay.
Till I see that, I'll be like, the fact that I can just purchase it, like a chemist can make it.
Yeah.
And at least for like RNA viruses, there's so many different varieties, but I've studied RNA viruses specifically since I started. That's the only, so I'm very RNA centric, and just the concept of like, just go and buy the sequence and give it to the cells and they'll make it. Like, it really is that.
It feels more like
really
a compound.
It feels like if it was true life and we can purchase it and lyophilize and overpowder and add water to reconstitute it. Like, is it really? So
sea monkeys.
This is the first answer to this question that has actually shaken my very adamant belief that viruses are alive. This is like the first time I'm like, maybe not.
Oh, something to think about. There you go.
There you go.
But here's an interesting thing, though. This is a question from our guest, somebody in the chat room earlier, Mike Shoemaker, who's asking: the common cold is coronavirus, and we have not been able to cure that, assuming because of mutations. Why would COVID be any different? But then, if it's mutating, if it's sort of evolving to, through natural selection and through mutation. That sounds like life on the one hand, but his question is why if we haven't cured that one, how come this one would be any different? And I guess somewhere in there, we have some coronaviruses that we've been able to stop. Maybe not the common cold one, but there have been some that we have. No. Or is it none of them? None of them. This is going to be the first one that we
don't have. We don't have any coronavirus vaccines.
None.
None.
Yep. And they either like circulate and then just magically disappear, or that's it. And
magic? That's what we're relying on?
That's really,
crossing my fingers and toes here.
When this started and people were like, well, this is COVID-2. What happened to COVID-1? And people were like, well, it just came. And like, that's it. Like, it just disappeared. Like, it's just, and so, virus is its host. So getting back to, the virus mutate, virus evolve. And so that is a very strong feature of what we define life. It can only do it in its host. So like I actually got that question from Twitter. Like someone messaged me like, can a virus replicate the genome in its own capsid and mutate there? And so that is like a big no. It cannot do it. And so a virus in itself, that genie trapped in a lamp cannot mutate, it cannot evolve, it cannot do anything. It is a chemical compound. Sorry, virologist, but that is a chemical compound until it has to evolve, because there's and that case is where this paradigm comes from, because it will then act like as a life entity, and it will mutate and evolve to cope with all the pressures that our bodies and our cells are putting to it, almost always randomly, but
To be fair, our own lives, while a virus needs a host, our own existence is dependent on being a host to all the microflora in our gut. So we have to be hosts in order to live. So is that really life? If you need all these helpers constantly doing all this work for you, does that really count then? Because by yourself, you would just die.
But what is life anyway?
Yeah, it is. It's all of it. It's all of it together.
It's
But to me, like, it's not whether they're alive or not. It's it's really what they're doing. And a lot of people just think of viruses as these negative things, and they just come and do their things and destroy our bodies. But not all of them are like that, or they can be, as we mentioned, repurposed to do similar things as we discussed for like plant viruses. There are viruses that have been quote unquote repurposed to destroy cancer cells. So like those are oncolytic viruses, and a lot of people don't know that oncolytic virus therapy is a thing, that you can design a virus that specifically targets a tumor. And so if you put that virus into our body, you can destroy a whole tumor. So like not all viruses are bad. There's viruses in the ocean doing their thing. There's viruses helping other organisms. Like
I know we're getting towards the end here, but one of the most fascinating stories I think that we covered on this show years ago was how infants don't have an immune system built up yet. And you're not born with a great amount of microflora, but up until about the age of 2, we have the highest viral load that we experience throughout our life. And it drops off as our immune system, as our microflora pick up and take over. But that our first immune system is children, as infants, is a viral immune system. It's going in there and destroying bacteria that could be invasive, because there is no other line of defense but those viruses to attack them. So in some ways that we are absolutely reliant on viruses for our immune system in the first place.
Yeah. So there's people tend to group viruses as, again, we are all very human centric. But there are viruses that we said infect plants. There are viruses that destroy bacteria, and there's some cases where those viruses are really good for us because they kill the bacteria that is bad for us. And so we just have to stop considering our species as the center of the world, because there's a lot of positives, and viruses just didn't come into existence to wipe off and just be the bad guys. There's
pathogens are an accident of nature. Pathogens are just like a one-off accident, miscommunication. Something's wrong. It's not intentional.
All a system. It's all a system. It's everything, using everything else, being a part of everything else, the ebb and flow of energy. I know I'm sounding kind of new agey here, but really, it's, trophic levels and molecules and electrons.
That is a really good question. And the last philosophical venture that I'm going to leave you thinking about is that I actually got that question one time. It's like, why our immune system has not evolved a way to like stop going wild and like in many cases killing us. Like it just goes so rampant in inflammation that we die. And again, if you then go up in the macro, it might be that it's killing you as the one host, but it's so that you don't spread it to the whole population. So again, just like in our cells, you might lose one cell, but you're saving the organ. It might be that in case of Ebola, for example, it might be that you just die so fast because our body just goes wild. But in reality, it's perhaps trying to stop it from, if you kill the host as a whole, then you stop spread in the human population. So it's species survival.
So really what we need to do, what we need to do is make the coronavirus more lethal. So that it kills people quicker before they can go and spread it. That finally makes sense.
We're not gonna do that gain of function experiment. No thanks.
I don't know. If it works, you gotta think about the herd. Not the individuals.
Yes. I heard you there. Efra, thank you so much for joining us tonight. I know it's getting rather late over there on the East Coast. Where are people going to be able to find you moving forward?
So I'm always, I want to say always, I mean always on Twitter. So they can find me at Naked Capsid. As I'm changing jobs, I am less so tweeting about viruses, but if anyone has any questions about viruses, my inbox there is open. I have a website that is linked there to my Twitter page too. And so you can click on there, there's a contact. If you don't have a Twitter and you want to message me, then there's a contact there with my email information and whatnot. I'm always happy to answer questions, even from what you may think of the most basic question in virology that you want to understand. Feel free to do so. So those are usually the two go-to places to find me.
Fantastic.
And you're venturing into more into communications now, and I think that's going to be a bright future, because this was fantastic interview. Learned a lot today. Thank you so much.
Yeah, no, thank you. Yeah, so I've been doing from Twitter science communication. So it's not my first time. I've been managing different Twitter pages for cell biology journals and stuff like that. So that's where I'm moving next. And if you think about it, every time that you're on social media, whether it's Facebook or Twitter or Instagram, and there's, let's say, the cell journal, and there's a tweet that comes saying the paper. Well, there's someone behind it that writes that tweet, that read the paper, wrote a tweet, and tweeted it. And for some journals, that's me. That's what I do. So that means I have to read a lot of papers way outside my area and like condense it. So that's the communication part of what I do now, and that gets me excited because like every day is a new discovery that I get to summarize and share with the world. So
yeah, that's exciting.
And as we talked about earlier, there needs to be more sharing of the science with the world so that we can get that trust up, right?
Yeah.
More sharing, more communicating. Thank you so much for your time and for joining us. We have more stories that are still to come. Will you be staying with us, or?
I can hang around for a little while. Yeah.
Okay. Fantastic. All
I'll
right.
duck out for a sec. I'll be right back.
You'll be right back. Okay. Well, we never stop. This show is going to keep going right now. Oh, yeah. That was a fantastic conversation. And we will have the links to Dr. Rivera-Serrano's information on our webpage. Thank you all for listening to TWIS. You are the reason that we are able to do what we do every week. That's right, bringing you the science, down-to-earth views, the news, a somewhat sane perspective, and hopefully we combat a little bit of that misinformation that is out there by bringing you some credible information in the world. If you would like to be a part of bringing more of this sanity and science to the world, head over to twist.org right now, click on the Patreon link, and choose your level of support. Be a part of bringing the science to more and more people. Thank you. We can't do any of this without you. Thank you for your support. All right, Blair, is it that time? The
It
time
sure
in the
is.
show where I play the theme music for Blair's Animal Corner?
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have a story that's a little early for Halloween. Bear with me here.
All right, I'm ready.
A wild story. I spent a lot of time on the show trying to demystify snakes and talk about how they're not scary. But I have one particular snake that I'll go ahead and say that it turns out they're very scary. This is a snake from Thailand. They're called the small-banded kukri snake, and they have been documented on 3 occasions doing something we have never, ever seen. Snakes normally, they swallow their food whole. They either constrict or they will inject with venom, and then they have these kind of backwards pointing teeth and they'll swallow their food whole. They're not chewers. But on 3 occasions, this snake was found cutting open the abdomen of a large poisonous toad, inserting their entire head into the abdomen of the toad, and then pulling out the organs one by one to eat them while the toad was still alive.
Is there a video?
What?
I do not have a video. There's a picture, but it's quite graphic. I'll just say, if you're listening or watching,
if you're watching the video, we now have a frog or a snake with a frog on it, wearing a frog as a hat.
Right. So this was seen from the Asian black spotted toad, which, as I mentioned, is poisonous. So it secretes a toxin from their parotid gland, which is kind of behind their eyes, kind of near their neck. If a toad even has a neck, kind of just head. But anyway, so they secrete this poison. So there's now 2 theories as to why these snakes might do this. One, it could be that this is their way of avoiding the toxin. So they are cutting open into the toad to avoid the poisonous secretion on the skin and go straight to the vital organs.
That would make sense. Okay.
Theory comes from a 4th observation in which they saw a snake actually eat a toad whole, but the toad was much smaller. So either the toxin isn't as potent when they're small and it gets more potent as they grow big, and that's why they couldn't swallow these bigger toads whole, or they were just too big to swallow for these small snakes. And so I guess they're getting real desperate. I don't know. But yeah, so there's something weird going on here. Again, this is only 4 total cases that were observed, so it's very, very preliminary. But it still is something that just has not been observed before and kind of shakes some of the expectations that we have for snakes and the way that they eat food.
I mean, it's really different from what we normally expect. The whole I'm swallowing my prey whole thing.
Yeah. For those of you that are now terrified of the idea of the snake that eviscerates and disembowels, they are harmless to humans. We are way too large. They are tiny snakes.
So far.
The one thing, though, is if you were bitten by one of these snakes, you'd have to wear a bandage for a pretty long time because they have a really powerful anticoagulant that they inject into the bloodstream when they bite. And
so you would just bleed to death.
You wouldn't bleed to death. You put on a Band-Aid, you wrap it kind of tight. It would eventually coagulate. We're big enough. It's fine. But if you were a toad, it's helpful because when they spend this extended period of time with their head inside the body of the toad, they don't want clotting to occur. So this kind of helps allow them to get to all of those organs.
Yeah, it's something that I do not want to see ever on a hike.
Nope, never.
So just beware if you're ever hiking in Thailand. Just if you see a toad on the ground, just assume it's a friendly, happy toad and be on your way. Don't take a closer look, is what I would suggest.
Wow. Yeah. Okay. So that's a new thing.
Yes.
Yeah. Mm-hmm. I'm traumatized slightly.
Well, let me now bring you to something fun and exciting, which is fireflies.
Okay. I like fireflies.
I love fireflies. California girl, didn't really go to the South at all growing up, and so I didn't see fireflies until I was an adult. And I was visiting my friend in Atlanta, and I just saw something out the side of the car when we were going to dinner. And I was like, pull over! And I jumped out. They're amazing. I'm so fascinated by fireflies. They're magical. And this is a study looking at fireflies in Great Smoky Mountains National Park. And a very particular thing that fireflies do, which I did not know, which is in this national park, thousands of male fireflies will congregate and they will flash together, synchronized.
Is this a mating thing?
Great question. So this was studied by the Department of Computer Science and Biofrontiers Institute at the University of Colorado Boulder. They wanted to see how this synchronization happens, how they manage it, and why they do it. It is definitely related to mating. It's a mating display. But are they doing it because it's just part of the way that their body functions? Is it an internal rhythm, or is it something that they do to communicate with each other? And they actually got to use 360-degree cameras in order to kind of see this from all different areas. And they also had a pop tent so that they could catch all the fireflies and then release them one at a time to see how the kind of the flickering display changed depending on how many fireflies were out. And they don't behave the same when they're alone versus when they're in a big group. So that definitely means it's some sort of communication. It is not an internally wired rhythm.
Yeah, like the temperature gets to a certain degree, and then that leads to how frequent the things are. But no, that was eliminated.
Yes. So so they have a few quick bursts, and then they have a several second pause, and then more bursts, and then a pause. And they say it looks kind of like a wave of light passing over the hillside, which sounds beautiful. I would love to see it someday. And so it seems to be kind of a competitive edge between the fireflies. So as they kind of all start to flash, they're big copycats. They all kind of do it together to try to match,
outdo one another.
Exactly.
Yeah.
Kind of, I was going to say match wits, match lights with each other when they are competing for a mate.
It's probably, it sounds kind of like it's like the firefly version of lekking, where the birds, the male birds all come around the female, and they're like all of them displaying and showing off.
I'm gonna puff out my chest. Ooh, I'm gonna puff out my chest too. Oh, I'm gonna move my head back and forth. Oh yeah, good idea. I'm gonna do that too.
Yes,
but
got that.
So part of where this computing department comes in from this study is they actually hope to use this group dynamic to help design swarming robots, which sounds terrifying.
I had no idea you were going to go from fireflies to robots. Okay.
It sounds very scary. I'm not necessarily with it, but I'm sure there's a good reason to have swarming robots. I just can't think of any right now.
No, if you have a large number of robots that are acting autonomously, or if one of the robots has instructions and the other ones can act based on what they see the other robots around them doing, then you can have larger groups of robots acting in concert. Then you could potentially, it's easier than controlling them all from a distance.
Right. Yes. I'm thinking of the latest Spider-Man film. I don't know if anybody saw that, but it's where they had a bunch of drones kind of working in concert to make holograms and things like that. And definitely, they reminded me of bees for sure.
Yeah, I just imagine swarming robots in outer space. That's just where I imagine them be. Sure. I mean, I know there are lots of uses for them down here on the planet, but I just think of them, swarms of robots in outer space.
Yeah, from strobing fireflies in the bayou to swarming robots in outer space. They're all connected.
Or we could replace those cardboard cutouts of fans in the stadiums with robots that, couples start cheering for a play, all the rest of them cheer. Somebody starts booing. It's just
pretty good idea. I like that. You should trademark that immediately.
Yeah. Goldizator in the Twitch chat room is saying, if one robot fails, the rest of the swarm can continue. Smaller robots are simpler and more reliable.
There you go.
Yeah. I was going to say, this is This Week in Science, if you just tuned in. And if you like the show, do you want to help us grow? Get a bigger audience? We would like that. Get a friend of yours to subscribe to TWIS today. That would be just wonderful and very helpful. Thank you.
Yeah. And if they happen to just leave their YouTube open, just browse over to This Week in Science, hit the subscribe button for them. And then it'll be like a nice surprise. It'll start popping up in their feed. They won't know why, but if they enjoy the show, they'll stay.
There you go.
Justin, tell me a science story.
Oh, I probably have one of those.
You should.
If nature communicated better, it may have if it had a newspaper that it put out, a newsletter, Twitter account of its own. One of the most prevalent content subjects that would be trending would be about who is eating who currently in the world of nature. Scientists at NOAA Northeast Fisheries Science Center have developed such a source of who is eating whom off the northeastern United States oceans. The predators were divided into 48 predator-sized categories and 14 prey species. Fish predators included, but are not limited to, Atlantic cod, Atlantic herring, haddock, goosefish, pollock, spiny dogfish, winter flounder, and the yellowtail flounder. Prey species included such fish food favorites as squid, zooplankton, shrimp-like crustaceans, shellfish, brittle stars, sand dollars, and sea urchins. Among the study's findings, most of the fish responded to changing abundance of prey by switching prey. They went through a sort of learning period when the prey in the highest abundance began to be targeted. That response helped do a couple of things. First of all, it meant that they had the most stable food source, but it also then helped stabilize the prey populations. So there's sort of this equalization thing taking place within these fish where they would because we were talking about the birds earlier, how the birds, if they lose their food source because of global warming, the clutches can die from starvation. In the fish world, they would just stop eating insects and go on to berries or something else. They would just sort of, it looks like they have this, and it's very interesting. They just go, they will change. We think of predators as often being sort of very specific feeders on specific things, but it's plastic. They can change. So this is actually a pretty huge study. This goes back to surveys data that was collected from back into the 50s and 60s into the 70s. So this is a pretty comprehensive long-term study. Really interesting. The findings are published in Fish and Fisheries, and it looks at these 17 species of predator and prey. Why is it that nature can find these balances so simply? And humanity can't sort of sort this out. Like, how come fish are so much smarter?
Yeah, I mean, it's something that when we had differently modeled societies, there is historical evidence that we were better at switching prey species and switching our resources based on seasonality and the environment that we were in. Native peoples are known to have really used fisheries very well and actually helped certain fisheries to thrive in their management and use of those resources over time. So yeah, I think it's more a modern conundrum, and maybe it's something that's based on how many people are on the planet, because we've gotten to this point where it's just really hard and we're all in cities, and how do we manage all this stuff? But I think nature and humans, as part of that, historically we've done a good job, but we aren't anymore.
I mean, how many times on the show have we said, like, oh, we thought we knew what this animal ate, but it turns out, we found out that deer sometimes eat meat, which blew my mind when we figured that out. So yeah, I think it's exactly that. Nature's a lot more flexible than we usually get.
However, Jane Goodall was on the Daily Show with Trevor Noah recently.
Yeah.
And one of the things that she sort of pointed out was that, like, if chimpanzees were as intelligent as humans, they would likely be just as destructive to the environment. That desire to take on a resource when you can control it is just too tempting for most life forms. But I do like this study for another reason too, because we've talked about how fish are going to have to be migrating as ocean temperatures change. This is a really good sign that, at least based on the northeastern fish species in the Atlantic, that there is the ability to change strategies, that there is ability to shift to the abundant food source for the predators. Hopefully the prey species can be as agile in shifting what they're eating. Otherwise, the whole system's gonna fall apart. But that is at least good news for fish populations in the face of global warming and changing ocean temperatures. As their population of fish that they encounter changes, strategies might be able to keep up in real time. So that's fantastic.
Yeah, hopefully with climate change and all the other pressures that are out there, it'll all continue to manage. But maybe we'll get better at managing. Come on, humans, sustainability's fun.
Yeah, it is.
It is. Didn't you have a story about Neanderthals?
Oh, yeah, I've got that too here. Let's see. Yeah. This is so there's been some constant improvement in molecular technologies that have allowed the scientists at Max Planck Institute of Evolutionary Anthropology to, in the past, determine with some high quality sequencing genomes of several Neanderthals, which then led to the discovery of the other extinct humans, the Denisovans. Now they've gotten to the point, they finally basically got the Y chromosome looked at in Neanderthals, which is not something that we've had before. And it's reinforced some hints that they had gotten previously about modern human Neanderthal interactions. But by comparing by comparing the archaic human Y chromosome and the Y chromosome of people living today, the team found that Neanderthal and modern human Y chromosomes are more similar to one another than they are to the Denisovan Y chromosome. Does that mean that we are all the descendants of Neanderthal? No. It actually means something crazier, that some modern humans around 370,000 years ago contributed heavily to the Y chromosomal lineage of Neanderthals. We've had a couple of hints of this before. There have been some hints that there was intermingling with modern humans and Neanderthals around the 200,000 year mark. This is putting it at around 370,000 years. That modern humans contributed to that Neanderthal lineage. So this is, is by well now established that all peoples of non-African ancestry carry a small amount of DNA as a result. That there's just one incident, or the most of that mingling was thought to be in that 50,000-ish year ago range. But the Y chromosome sequence now provides evidence that Neanderthals and early modern humans met and exchanged genes before the major out of Africa migration, potentially as early as 370,000 years ago, certainly more than 100,000 years ago. What's kind of also interesting about that is that's putting it in a time frame earlier than we think of modern humans as being modern humans. So there's still so much that we don't know about how modern human Neanderthal interactions took place, but it is more and more evidence that within the Neanderthal lineage itself, there's a good braided stream, to the point where it makes you then start to pause a little bit about what is truly Neanderthal DNA that we've looked at as Neanderthal, and is co-contribution from modern humans. How much is that we attribute to being Neanderthal genes could be archaic modern human genes. It gets very, so what we really need is a better look at Denisovans, because the Denisovan Neanderthals are much closer aligned in many other ways. So that's the part of the puzzle. When we get the Denisovan aspect in there, it may be that the Neanderthal is more of a hybrid of Denisovan and archaic modern human than it is its own branch of humanity. Lots of fascinating, interesting things that are going to come from this going forward. But
yeah, I can't wait to see, as we get our technology improves and our ability to look at these questions, what kind of answers we will find about what how the braided stream really, how it was really braided, how all these ancient lineages came to mingle, how, which lineages died off and how, and what genes were contributed.
And part of the noise in that is also being able then to separate what's a Neanderthal from what's a human. I got sort of an anecdotal example.
Humans, what are they even?
Very good friend of mine is Scandinavian. The lineage of going back for as far back as you can look of being Danish. Didn't travel much. Did the 23andMe, came up with 12% British. Well, that's because likely there was some Viking ancestry that left and went to Britain at some point. So then you have to say, well, is that 12% British genome, which they identified amongst British people, actually from Britain? And then, and it only depends on how far back you put the slider. Are you looking 300 years ago? Are you looking 500 years ago? That all, changes who's from where anyway. So it's all kind of ridiculous at some point. But now we have to start looking at how much of our DNA genome is just current modern human that also was in Neanderthals. Anyway, it's a lot of fascinating fun to come from this.
I love it. Neanderthal, Y chromosome. It's ours. It was the humans. Let's talk for a second. I've got 2 stories before we close out the show. Let's talk about brains, because I love the brains. So in a new study published in Nature this week, researchers at the Riken Center for Brain Science in Japan have described an area of the brain that is called SUM. SUM. This is an area of the brain in the hypothalamus that is related to memory formation, specifically involving social novelty. Now, what does that mean? That means meeting new people. Well, in this particular situation, it was meeting new mice, because they were looking at mice. In the study, they took mice and gave them different kinds of novelty. They either got contextual novelty, so a new running wheel in their cage, or a ball, or new fluffy bits to play with in their enclosure, or they got social novelty, and the social novelty was exposing them to new individuals who they had not met before. The SUM they recorded from and discovered, they looked at brain activity in a large area of the brain, but focused in on this area because they found its activity was much, much higher when social interactions were new, when there was novelty there. Was not as active during the contextual interactions, or if they were meeting a familiar mouse friend. So what they did that really dug into this to discern how it works, of course, there was some manipulation involved. So because we have optogenetics now, this wonderful tool involving blue light that allows researchers to turn neurons on and off, they used optogenetic light stimulation to stimulate the areas of the brain that SUM connects to. And they found that when it was active, it was connecting to a memory area of the brain known as the hippocampus. It's involved in consolidation of new memories. And so when a new interaction, new experience happens, the neural activity is going to put connections into the hippocampus, and then the hippocampus is going to take that information and send it out to other areas of the brain to make sure that the memory turns out becomes a long-term memory if it needs to be, so that it can get stored. And there were 2 areas of the brain involved. When contextual novelty, the new running wheel or a ball or whatever was involved, there's the dentate gyrus part of the hippocampus, and that's kind of the part of the hippocampus, it's on the way out. That's like information goes into goes into an area of the hippocampus called CA2, and then it moves out through the dentate gyrus, and then it goes out to other areas of the brain. So contextual novelty stuff was connected to the dentate gyrus. Social novelty was connected to the CA2. When they used optogenetics to turn on or off the dentate gyrus or the CA2 connection, they were actually able to make the mice behave as if they were having a new social interaction, even if they weren't. So they turned on that SUM to CA2 connection when mice were with a mouse that they'd been friends with for a long time. And instead of going like, "Hey, Fred, what's up? Good to see you again," they were like, "Oh, pardon me. Would you happen to have any Grey Poupon?" They acted a little bit differently because it was a new social interaction. And so by... Sorry, I don't know where that came from, but...
I believe the 90s is where that came from.
No, it might have been earlier than that. It might have been much earlier than that. For the kids listening, Grey Poupon is a form of mustard. And there used to be a television commercial where somebody would approach a stranger and ask them if they happen to have that brand of mustard on them at the moment. And to which, of course, they always did, because who doesn't carry mustard with them wherever they go?
And I love asking strangers for mustard.
Yep.
It's on me.
It's how the world used to work.
But anyway, by doing these manipulations, they were able to confirm that mice have an area of the brain that is specifically involved in meeting new mice. And humans may have an area like this as well, that may be involved in helping you through those new social interactions so that you can remember new individuals if they are going to be important to your future dealings. Right?
I need to figure out how to flash that portion of my brain with light in just the right way to actually remember people's names when I first
yeah, I think that's a completely different area of the brain that just has like the magic erase marker in it always.
Yeah. It's like, oh, nice to meet you. I immediately forgot. Can you tell me your name again? Oh, and it's gone again. Sorry.
I'm just glad that there's finally a therapy that might be developed for introverts.
Yeah, well, maybe. I don't know if that's what it's good for, but yeah, potentially. And my final story for the night is bird brains, because you know how much I love the bird brains, not just brains, but bird brains. And we have talked a lot on the show about, how birds think and how birds have much smaller and, from an anatomical perspective, what anatomists would call a much simpler brain. We have a convoluted cerebral cortex, which we have always thought to be responsible for our higher cognitive functions and for things like consciousness. But we have a really hard time asking other animals whether they're conscious. How do you go about finding out if another organism is conscious of their day-to-day experiences? Well, ta-da! Science! Researchers have figured out a way to start getting at this question with carrion crows. It's not tickling. No. It is not tickling. Yes. So they looked at, it was a very small sample size. They trained 2 carrion crows to do a very specific kind of task. It's a delay. It's a, what was it? Let me close the window that I was just opening so that I can say exactly what the type of test was. Well, it was a delay. This doesn't really matter what this piece, a delayed stimulus detection task. This is what it is. Delayed stimulus detection task. But what they wanted to show was that there is a subjective part to birds' perception, and that there is an area of the brain, to confirm that there is an area of the bird's brain that has the capabilities that we ascribe to our prefrontal cortex. So the area of the brain that they looked at is an area in the back of the brain. So we think of our prefrontal cortex, it's up in the front of our brain. The neocaudolaris. It's an area of the bird pallium, which is a laminated structure. It's not convoluted like the human cortex. It's very smooth, but this area in the back of the brain is thought to have the same cognitive capacities of perception as our prefrontal cortex. So they put electrodes in there. They trained these birds to look at a mark on a screen, and then they trained them on a yes or no task. So if they saw the light turn on, the mark turn on the screen, and then there was a delay, and then they were trained to respond a particular way, yes or no, if they saw another certain stimulus. So it was basically like, hey, here's this box. Now, wait a second. And now we're going to tell you what you should answer. The part of telling the birds what they should answer was to remove randomness, and that the birds might just be learning some kind of trick to the answer. So by telling the birds, this is what you have to do, they were learning the strategy and learning how to play the game, but they had to rely specifically on what they had seen previously before the delay. So They knew exactly what the birds were seeing because of the electrodes in the back of the brain. It's like, oh, if there was a box there on the screen, visual cortex information went into the brain lit up, and it was like, yes, it was there. If it was not there, there was no activation in that part of the brain. And if it was shaded at somewhere in between 100% brightness and 0, it would, the neurons would light up somewhere halfway as well. So then they said, let's do this thing. Let's do this test. And they had them either see the box or not see the box and react in the way that they were supposed to react. And because of the way that they set the test up, they were able to determine that the birds were not just visually responding to a stimulus, but actually subjectively aware that the stimulus was there, conscious of its presence, and making a choice to click their answer, to click the response box. Because there was that delay, the birds were taking their perception and acting on it in a conscious fashion. And so this study, they say it confirms that carrion crows, have consciousness. Now, okay, lots of animals. You look at the Blair, Efra, you have your dogs. I've got my cats. I look at my animals, and I've been an animal biologist for years. I'm like, but I still look, my cat is conscious. My cats know their name. They're not just doing tricks. They know what's going on, right? But they're also mammals. A bird is a completely different, it's a different group of animals. And if consciousness is something that we share with birds, that means consciousness itself goes back to an ancestor sometime before the split.
Yeah, it could be convergent. But
or it could be convergent. It could be convergent.
I tend to agree with the first premise.
So now the question is, if we look at this, does this mean all birds are conscious? Does this mean that if we go back, then the crocodilians, are they conscious? Like, how far back does it go, and what areas of the brain and what brain designs, what structures, and how does the structure lead to the function? What amount of brain and wiring, networking within it, allows consciousness?
Yeah, there's consciousness. I feel like there's lots to that, right? There's like a sense of self. There's like an understanding of your impact on your environment or your population. So then if you start thinking about animals that live in cooperative groups, there's been lots of study on lots of different kind of levels of organization in the animal world about how, especially with altruism, like animals that help each other in groups, if an animal is better at helping other animals, they get helped more. And if they're not as good at helping others, then they get helped less, which also kind of implies a sense of self and a consciousness, because you're not going to help me, so I'm not going to help you.
Yeah.
Which indicates an understanding of your own self. So I feel like this is one of those things that the more you pick away at it, the harder it gets to find a hard line in between this consciousness and not.
My favorite
maybe there is no hard line. I mean, maybe it is a gradation. I mean, yeah.
My favorite example would be learning and the act of teaching. Like, I'm sort of picturing orcas doing things, or dolphins using tools or using strategies to attack prey that then the young observe and then repeat. So there's and especially with the orcas, just because I've been watching too much orca stuff lately, reading too much orca. Yeah. They will do sort of faux hunting where they allow the young to sort of take the lead And then we'll kind of then show them how it's done and then let them go do it and then show them how it's done and kind of go let them do it. And that's both an awareness of one is learning. I have to pay attention to this other individual and try to recreate what they do, but also that awareness of I'm going to illustrate. I'm intentionally going to demonstrate and then allow my pupil or offspring to attempt to replicate and see how they do. And then I'll show them again. And so that sort of exchange, you know, I'm picturing this as I'm trying to come up with examples, and they're all mammalian, and then there's birds. Blair, can you think of anything? Is there a crocodilian thing? Because crocodilians can do a group hunt.
Yeah, and we had a really cool story so many years ago now, where the kind of the bigger crocodiles would sit and wait, and the smaller, more spry, faster crocodiles would help chase prey to that crocodile that was waiting. So there's teamwork going on.
Mm-hmm. Yeah. So I mean, when you're talking about animals that exist in social groups, there's, I mean, definitely awareness. There has to be some kind of conscious, I mean, maybe it's not conscious, but this study suggests that, you know, this is much more widespread than we have the evidence to think or to have thought before. And that there's conscious awareness of self and other, like you were talking about, because you have to be able to exist in that group and work with others to exist in that group. And if you're not, you know, not aware and conscious, but awareness and consciousness are like two different things,
right,
psychologically. Right. But I think it gets, it's just a very interesting question. The idea of being conscious of a perception, to be able to hold on to it, make a choice based on it, and act on it. There's a lot. There's a lot in there.
I mean, I have another. Today, I was talking to a classroom of students about bearded dragons and how they like wave at each other to be like, "Hey, this is my space." And you know, if you're a smaller bearded dragon than that big bearded dragon that waved his hand, you're not going to challenge his space. So how do you have enough sense of yourself to go, okay, I'm smaller than that dragon, and that dragon just signaled to me that that is his space, so I'm going to leave him be and try to find another space. Mm-hmm. There's just, it's, if you keep picking at this scab, it gets harder to identify.
Why do you have to put it like that? There's two more examples I'm going to leave to you.
Satisfying
but we want to end the show.
I know we're going to end the show. There's two videos that people should try to find on the YouTubes. One is of an otter and one is of a penguin. And in each of these cases, these animals are in the water when an orca comes by and leap onto a boat to avoid being eaten. Now, they're in close proximity to humans, which normally they would do anything to avoid, but they're jumping on. Well, the penguin is just basically jumps into the boat with a bunch of people and is like, "Hey, how y'all doing? So what are we doing? Are we fishing? Are we sightseeing? Because it looks like a nice boat you got here." And it's just like sort of like really trying to ingratiate itself with the group there as quick as possible, as this orca is like coming up and like looking like, "Where'd you go? Where'd you go?"
I'm just looking like a human over here. That's all. Doing my best to fit in.
Aside from the natural fear of a predator driving this, there is also some, okay, that's bad, but this isn't as bad. That's much worse. And I'm going to take care of myself by joining this and avoiding that. I think this, it's really hard to look, and that's that's a mammal and a bird. It's really hard to look at the animal kingdom and really think that we are that different. Sometimes people forget, and it's worth reminding every once in a while.
Are you going to say we're animals?
Yeah, we are. Yeah, we're not really that.
No, I love telling that to a classroom of kids and them all going, "No, we're not."
Yeah.
Yes, we're mammals, so that's an animal.
Shouldn't be so strange.
Yeah, I mean, our parents tell us. I mean, I tell my son, "Don't act like an animal," I am one. I can't help it. Okay. I think that does it for our show for the night, fellow animals. We have scienced and we have made it to the end. Thank you for listening. I hope that you enjoyed the show. It's time for those shout outs. Want to shout out to Fada for helping with social media and doing the show notes. Thank you so much. Gord, thank you for manning the chat room. Identity4, thank you for recording the show. And as always, I would like to thank our Patreon sponsors and the Burroughs Welcome Fund for their generous support. Also, Efra, Dr. Efra Rivera-Serrano, thank you so much for joining us tonight on the show.
No problem. Thank you for the invite.
You're welcome. It was great. All right, time for me to read those Patreon names. Donathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Josiah Zahner, Mark Shoemaker, Sarah Forfar, Donald Mundis, Gerald Sorrell, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred SS104, Corinne Benton, Sky Luke, Paul Ronovich, Ben Bilgnell, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Raleigh, Mark Hessenflow, Gene Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brandon Minish, Melazon, Johnny Gridley, Flying Out, Richard Porter, Christopher Dreyer, Mark Mazaros, Artyom, Greg Briggs, John Atwood, Robert Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Greg Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Alex Wilson, John Ratnaswamy, Sue Doster, Jason Olds, Dave Neighbor, Costi Ranke, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Ben Rothig, Gary S., Ed Dyer, Tony Steele, Ulysses Adkins, Brian Condren, Jason Roberts, and Dave Friedel. Thank you all for your support on Patreon. And if you want your name to be read at the end of the show, you can find information at patreon.com/thisweekinscience or just click that Patreon link at our website. And on next week's show, we will be back once again. Mm-hmm. When will we be back, Justin?
We will be back Wednesday, 8 p.m. broadcasting live. That's 8 p.m. Pacific time. Broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Hey, I know you love looking at our beautiful faces, but if you want to multitask and you want to listen to us as a podcast, just get the audio only. You can search for This Week in Science wherever podcasts are found. If you enjoyed the show, be sure to get your friends to subscribe, grab their phone, hit that subscribe button as well.
For more information on anything you heard here today, show notes and links to stories will be available on our website, www.twist.org, and you can sign up for our newsletter while you're there.
That's right. You can contact us directly as well. You can email Kirsten at thisweekinscience.com, Justin at twistmedia@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will end up, I don't know, inside a capsid in a test tube in a lab, and we'll never see it.
Oh, no.
You can also hit us up on the Twitter where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes during the night, please let us know.
We'll be back here next week. Oh my gosh, it's almost October. And we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just get understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Oh, yeah, we've come to the end of the show. No, not Elect More. Oops. Doom, 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 doom. Not more Scientologists' strengths. No. Happy Octoberween! That's right. Tomorrow it's October, and I know I haven't done things that I should have done already. And why does it keep going? I'm having issues getting things done. What?
Same.
I keep wondering, seriously, it's like people are running at this breakneck pace to continue to make things normal. They're like driving themselves to oblivion with normal distractions, and I just am tired, and I'm feeling like I do need hugs, Thunder Beaver.
My hips hurt. Don't mind me.
Oh, I like the bumper sticker theme that you designed Identity for. That sounds great. Oktoberfest. That sounds funny.
I'm looking at the Fat Bear bracket right now that somebody.
Oh, Fat Bear brackets. We should look at that as a group. If you don't know about the Fat Bear,
know about this, and I'm obsessed with it already.
You didn't know about the Fat Bear competition? No.
Wait,
Fat Bear Week.
The National Park Service, their social media team is really good, but also they do fun stuff. They don't take themselves too seriously, and I love it.
Yeah, so, okay, here I'm going to screen share. For those of y'all who don't know, Fat Bear Week. Fat Bear Week is the competition to choose the fattest bear. Mm-hmm. Thanks, Noodles. Yes. Yes, and so there is a bracket. So there's a single elimination tournament for each set of 2 bears. Vote for the one you think is the fattest. The bear with the most votes advances, and one will be crowned. So, okay, so we've already got a couple of days worth of polls. So here's the bracket. There we go. Fat Bear Week bracket.
So today's
there's-
closed. I guess they're on Eastern time.
Yeah. So you can download a bracket, make your own predictions for who the fattest bear will be.
402.
So so we have, let's see, check back for voting results. So we haven't gone very far, but we've gotten
the one at the bottom.
ooh,
151.
151 got chunk.
Yeah.
Yeah, so these bears are like, how ready are they for the winter? They're chunking up for their hibernation.
Those are some good boys and girls.
I think Justin, I think you're right. The one, I don't know, this one at the bottom's pretty big, but the one at that one there, 151.
Mm-hmm.
But the 856 bottom one also looks. That's a good competition right there. But I think the one at the bottom, but that one it is a bear rump shot. It could be the angle.
Bear rump.
I got a couple of questions in the chat room here. One is, what am I doing and where am I? I ask myself this every morning. Actually,
are we supposed to answer this question, or is this for you, Justin?
That one's for me. Where am I? What am I doing? I'm currently in Denmark. I am taking classes remotely and looking to be re-entering the workforce flow thing sometime at the end of next month, perhaps. But yeah, I've been taking a pandemic-cation to a place where there's less cases and frequent testing available. It's Denmark is nice. If you ever get a chance to travel anywhere again, this is not a bad place. Also, let's see, random genre talk. I have a question for Justin in his study at the beginning about the matter percentage of the universe. Do black holes count as matter or energy in this calculation? So that's a great question.
What about the geodes that we were talking about? Those are matter, right? But it's dark energy.
That's the dark energy, which is the mass, the predominant form of energy. So as far as the black holes, I don't know how exactly they would account for this. However, the study was based on observing galaxies. All right. So if you have, I suppose, if they're galaxies have the most massive black holes at their core, so that matter and energy should be accounted for just by the fact that you're looking at these galaxies, because at the center of each of them is the biggest form of black hole that we know to exist. However, I don't know about the free-floating black holes that might not be associated with the galaxy that maybe took up everything that was around it. But I don't know how, when we look at that spread of matter, the way it's, what am I trying to say? The words with how it's sort of all connected into the threads and webs of matter that is dispersed throughout space. Now, I would assume the black holes would be there because they need the matter to begin with. So they should be in and amongst those galaxies clusters. So based on that, I would say that that's how they've accounted for black holes as the matter energy. But that calculation they were doing was matter as energy. So I don't know do black holes count as matter or energy in the calculation. I suppose it would be both.
This bear's number is 747, and is that because he's a jumbo jet?
Yes. It kind of is. That's another rump shot, largely, though.
It's a rump shot.
It's sort of.
Let me show you my favorite. I'm Holly. Holly is my fave.
Oh, look at her go.
Oh, Holly! I don't even think Holly can walk. I'm just gonna hibernate right here, folks. That's it.
Yeah.
I'm done.
I
What
love
did-
you, Holly. You're my
What
favorite.
was Holly getting a hold of? That's a lot of salmon.
Seagulls.
Seagulls.
No, the seagulls are hanging out.
Other
'cause-
bears.
I know. They're like, Holly is a good source of food. I will
picking
stay
it up.
Holly
here.
has been eating other bears, no doubt.
What's
Yes.
sweet? Oh.
And then, yes. The looking at bears and listening to stories about matter in the galaxy. Yes. It all makes sense now. These tendrils of connection. No, Shubru, we're not talking about the debate from last night. Nope.
Nope. No want.
No want. That's why we like fun science.
Well, I wouldn't actually.
And bear rumps.
I wouldn't mind talking about the debate.
Nope.
I really wouldn't in the after
Nope.
show, but there
Nope.
wasn't one.
I know.
Yeah, that's the
So,
one thing I'll say.
I don't
is
really
like
have-
the commentary of I don't know who won that debate, but one thing's for sure: America lost.
Yeah, I don't. Yeah, I don't think that the one happened.
Yeah, it didn't. Lots of shouting
so-
occurred. I didn't even watch it.
Hey, what?
Yeah.
Yeah, exactly. What? I don't know. Fat bears. That's all I know right now. It's the most important thing.
It's so heavy.
Oh. Oh my goodness. How's everyone holding up? All good out there?
Yeah.
All good out there in the world? I think, Justin, you are starting to get your coffee is working. Getting your
It
day
just
ready.
Stopped working. Yeah, it is almost 8 o'clock in the morning where I'm sitting. Had a pot of coffee, and I think I'm almost ready for a nap. Something's wrong.
That's like every week. Oh my goodness. This week I've been very tired. We've got some kind of a little cold bug in our house, and we don't know how we got it. But guess who has 2 thumbs and went to get a COVID test today?
Oh,
nice.
Well done.
How's your brain?
It's fine. They did not do the nasopharyngeal swab. They just did a nostril swab. Although they did, it wasn't, they went up there. Like, wee, let me get up in there.
So it's kind of interesting. I got tested here.
24 to
in Denmark.
72 hours. We'll see.
Yeah, I got. Oh, that's a good turnaround.
Yeah.
That's a turnaround I had. It was 24 to 48 hours. Yeah, I think we got it. The results within 24 hours. But it was a back of the throat swab. They did not put the. I was all like cleaning out, like before they did the thing. Like I don't want them to come out put the thing up and then come out with a big bogey.
Right?
Like, just be a little,
like, that's just
Although it
gonna
would
get
be
in
the first time, I'm sure.
I didn't want them to find anything weird in there. And then it was say and it went in the back of the, and that was the way the test was done here in Denmark.
When they did mine, they were like, "You will gag. Don't be embarrassed."
Oh,
Here
so
we
you got
go.
yours was also the
They
throat
did
test.
both. They did both nostrils and my throat. Yeah.
Oh, interesting.
All the
So
things.
This was back in June.
Yeah.
And I had all of the symptoms except
and you
for
said-
the only thing I didn't have was the dry cough. So,
but that's
I didn't have
what
any
it was.
symptoms,
It was just
but
a
I had
cold.
been on I had just
was a
gotten to the
virus.
country, so you have to get
viral
tested
infection.
at the airport when you leave the airport.
Yeah.
You have to go in.
The Brushwoods, Brian and Bonnie have COVID.
Oh no!
Yeah, Bonnie.
no.
Bonnie's having it worse right now. But they're making their way through that. Supposedly, hopefully, all their kids are okay. So they have 3 daughters, something like that. But yeah, I
hope
can't
they-
imagine
I hope
having.
they all start with the letter B.
Pull through.
I hope so.
Yeah. I can't imagine having a large family and trying to isolate and like deal with that and parent and homeschool and that would be quite a lot. It would be quite a lot. Yeah, their last test was negative. Yeah. Anyway, tests. We need more tests, everybody. Can we get more tests?
Gaurav Sharma in the chat room says he needs to go get his flu shot. Yes.
Get your flu shot.
If you are a person who can get a flu shot, get your flu shot. Get that flu shot.
Yep. I got mine. I got mine.
Yeah. I haven't gotten one yet. I might get one relatively soon. Here, though. Is it going to be the same flu shot? If I get one here?
Northern Hemisphere. It's Northern Hemisphere versus Southern Hemisphere, so it's probably the same.
Because people don't travel to different hemispheres?
Or yeah, I don't know. They travel within more?
Yeah. Most travel.
Right now, nobody is. Right now, nobody is. So it's probably, this time it would be perfect, but interesting.
Yeah.
I knew there was different shots, but I didn't realize how it was broken down.
It's seasonal, so the way that it works is because Australia and Southeast Asia, they have their winter, and whatever strains seem to really happen there are the ones that we expect will start to happen in the Northern Hemisphere when our indoor season starts to happen. Mm-hmm. And it's not that people don't travel back and forth. It's just that because of the nature of the virus, it is more seasonal. You can get the flu just about any time if you don't have if you don't have immunity for it. But it's more likely that you'll get it during the winter because of all the factors that are involved in allowing for the transmission of the virus. Humidity, cold, all that kind of stuff. Yeah. You there, Justin?
I am.
Your brain ended.
Did I literally freeze in real time? Sorry,
all three of us did.
Sorry, I missed it. I was. So this thing happens where I was reading something. I was reading something, and this has not been a problem in the past, but my older brain, if I'm reading, my auditory senses shut down. I can't hear anything. So I was reading something real quick, and then I completely lost what was no longer able to multitask. This is
aging.
Brain. This is what aging looks like. Were you saying something to me? I'm sorry.
No, not at all. Nope.
Good. Right.
No.
It also could just be COVID brain. See, I'm like a couple ticks behind now too. But there's lots of really good articles about how hey, even if you feel like you've found equilibrium in the time of COVID, you still probably have at least 10% of your brain that's constantly focused on the fact that we're living in a pandemic. And so it's harder to multitask. It's harder to get things done in a normal amount of time. It's harder to feel motivated. And it's because your brain is constantly busy with something it wasn't busy with before.
Mm-hmm. I really like the idea of just everybody taking a vacation for a month. Let's just plan for it and just, can we all please just not do for anything?
Yeah.
Just I don't want to do anything.
Only healthcare workers go to work. Grocery stores can be open 3 days a week.
Right. We don't need them open
every day. Plan ahead.
Just plan ahead. Yeah.
Close down for a month. This is the thing Justin's been talking about forever.
I know.
Yearly shutdown.
I'm not saying it for getting rid of the virus. I'm just saying so that we all have a vacation.
Oh. Yeah, it is weird how
We didn't
We're
get a
all
summer
stuck at
vacation at all.
home.
And yes.
But yeah, but
Going
still
anywhere.
need a vacation. But
Yeah,
can't
we
go
need
anywhere.
we need a do nothing November.
We're
Right.
distracting
Every year.
ourselves with work, and it's
Right, because now also
Yeah.
like people like myself who had an office, I have the lines between work and not work are blurring more and more and more. And at the beginning of things, I was like, okay, I'm going to work at 9, quote unquote, finish work at 5. Like very clear lines. Those lines are blurring more and more every day. And it causes problems.
It's like, that's cute.
Yeah.
Make soap November. Well, that sounds fun. Ooh, I like internal monologue short stories. Carol Ann, that sounds fascinating. Yeah, multitasking is a myth. That's for sure. It's true. It's true. People say we can multitask, but no, we can't. Not very well. Are we tired?
Mm-hmm.
Yeah, give the pint of blood today you might need tomorrow. Yeah, if people can go give blood, now is the time to do that. Blood shortages, and if you are able, that is something very helpful. No vacations on cruise ships. No, bad. Well, not right now anyway. Ugh. I would like the
Also
Lysol
generally bad
cabin.
because their environmental impact is off the charts.
Yeah.
So generally cruise ships, maybe we're done with them. Just
a sailing vessel.
Yeah, you could go on an eco cruise, which is like 50 people or less, and those are very cool. But those giant cruise ships, maybe they've had their time. Maybe they're good. Walking dysentery, ballast, invasive species delivery systems.
Oh, I miss movie theaters. That's the one I do miss a lot.
A lot of movie theaters are now doing private rental show times. So like I have a coworker who was able to rent out a theater for $99 for a showing, the entire theater. And so you can have up to 20 people, which like he's an essential worker. And so like he and a bunch of people who are going into work together every day and are already kind of in this pod are all going. Yeah. So that's pretty cool. But he invited me and I was like, well, I'm still kind of working at home. And I also have other, the other thing is I have a different social engagement this weekend. Yeah. And so I'm trying to have like one every 2 weeks, if that, right? So like, it's like, 2 days in a row with different social circles. No, I'm good.
Yeah. So this is a reality that I have declimatized from. Since being in Denmark, it is not there here yet. You could go to a restaurant. Mm-hmm. They have implemented face masks only recently at restaurants for when you're standing. You have to go in with one on, but then you take it off, of course, right away when you eat. But staff and waiters and stuff, these people are now wearing them. But I'm going to have to reintroduce myself into all of that, turn back on that part of the brain that's always thinking about this. Because I haven't been.
That's nice. That's nice. I would like to not do the thinking. Okay. I think that would be nice. I like the not thinking. I would like to turn my brain off for a while. Maybe with some sleep.
Oh, gotcha. All right. In that case, I think it might be time to say goodnight. Oh, I was too late. She's already asleep.
She's already sleeping.
Say goodnight.
Say good morning, Justin.
Good morning, Justin. Good night, Kiki.
Good night, everyone. Have a wonderful week. And we do look forward to seeing you again next week with more science. And I hope that you stay well. And, stay well, stay safe, stay healthy. Thank you for being a part of the night with us tonight, or the morning, wherever you are. Good night, sweetheart. Well, it's time to go. Boom, 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 boom.